There is a shark alive right now that was probably born before the United States existed. It's still growing. It might not have reproduced yet. Picture a gray shape sliding past an ROV light. So slow the camera reads it as drifting debris. You'd call it harmless. That's the mistake. Its eyes are being eaten by parasites. Its flesh can poison a dog. Its stomach holds seals it should never have caught. And the simple answer, it's slow because it's cold, explains almost nothing. Nobody has watched one mate. Nobody has ever counted them. Everything we know came from corpses. Pull a Greenland shark up through a hole in the ice, and the first thing that hits you is how little it does. No thrashing, no panic, no teeth snapping at the rope. It comes up like something already dead, a long gray body sliding onto the ice, skin the color of wet stone. And it just lies there. Breathing, if you can call it that. Barely moving, the people who have hauled them up describe the same thing again and again. The animal seems uninterested in its own capture. Now look at the size of it. This is not a small fish. A full-grown Greenland shark sits in the same weight class as a great white. Put them side by side as silhouettes, and the Greenland shark is thicker, blunter, less designed-looking, but no smaller. This is one of the largest predatory sharks in the ocean, and you have almost certainly never heard anyone talk about it. That's the first strange thing, and it's easy to skip past. We have named, filmed, tagged, dramatized, and merchandised nearly every large shark on Earth. Great whites have their own week. Tigers, bulls, hammerheads, makos. All of them have a public image. The Greenland shark has a nickname instead. Sleeper shark. Its scientific genus name carries the same idea, built from the Latin for sleep. Even the taxonomy is a shrug. Look at what that name does. It tells you the animal is slow. Slow tells you it's weak. Weak tells you it can't hunt. Can't hunt tells you it scavenges. And scavenging tells you it's a garbage disposal with fins, drifting around the Arctic eating whatever sinks. Every one of those steps feels reasonable. Every one of those steps follows from the one before it. And the whole chain rests on a single observation. It looked lazy when we dragged it out of its own environment. That's the problem. Not that the chain is obviously wrong. It's that almost nothing in it came from watching a living Greenland shark do anything in the place it actually lives. Because we didn't watch. We killed. For a long stretch of the 20th century, this animal was worth money, and the money was in its liver. Greenland shark livers are enormous and oil-rich, and shark liver oil had industrial value. Lamps, machinery, lubricants. So a fishery grew up around it in the North Atlantic. Boats went out, lines went down, sharks came up. This went on for decades, and the numbers taken were not small. Thousands of animals. Generation after generation of fishermen who could tell you exactly how one felt on the end of a line. Now here's the part that should bother you. That entire fishery taught us almost nothing about how a Greenland shark lives. Sit with that. We had a commercial operation built around one species. We had access to a steady supply of bodies. We had decades. And at the end of it, we could not say how they hunt, where they breed, how many there are, how long they live, or what they do for the roughly 99% of their existence that happens somewhere other than the end of a hook. We knew what the liver weighed. We knew the meat would poison you. That was the harvest. Think about who was doing the recording. A crew working the Arctic off a small boat is not a research team. They had a hold to fill and weather coming. Nobody was noting the sex of the animal, or its length, or the depth it came off, or whether its reproductive organs were developed. Why would they? It was oil. The shark was the container the oil arrived in. The data that would answer almost every question we now care about passed through human hands, thousands of times, across decades and went over the side. You can kill an enormous number of an animal and still not know it. That's not an accident of history. That's a structural fact about what a dead body can tell you. A corpse is generous with some things. It hands you anatomy. It gives you jaw shape, tooth arrangement, skin texture, the plumbing of the gut, the chemistry of the tissue. It gives you a snapshot with perfect resolution and zero duration. And duration is where biology actually happens. A corpse cannot tell you what the animal was doing an hour before it died. It cannot tell you how it found what's in its stomach. It cannot tell you whether it was hunting or drifting, whether it was alone or not, whether it had ever mated, whether it was 3 years from death or 300. The most important verbs in an animal's life, hunts, waits, follows, breeds, grows, hides, do not survive the animal. And the Greenland shark's stomachs were interesting. That's the part that started to itch. Cut them open, and you find things that don't fit the garbage disposal story. Fish, yes. Squid, yes. But also seals. And sometimes the remains of land mammals. Animals that have no business being in the water column at all. That must have fallen in or been swept out or died on the ice. A slow shark with a seal inside it is not a solved animal. It is a question with a body wrapped around it. But a stomach is still a corpse's testimony. It tells you what went in. It does not tell you how. It cannot distinguish between a shark that caught a living seal and a shark that found a dead one. That distinction is the entire difference between a scavenger and a predator. And the evidence we spent a century collecting is structurally incapable of answering it. Hold on to that. It comes back. And when it does, it gets much worse. So why not just go and look? Why didn't anyone put a camera down there and settle it? The obvious answer is the environment. The Greenland shark lives in water near freezing, often under ice, often deep, in a part of the world where a research season is short and expensive and the weather can end you. It's dark. It's remote. It's the kind of place that punishes curiosity. That explanation feels complete. It isn't. Because we have gone to worse places for less interesting reasons. We have filmed a giant squid in the open ocean, an animal that was a legend for centuries. We have put lights and cameras on hydrothermal vents kilometers down, in water that comes out hot enough to melt equipment. We have sat crude submersibles on the floor of the deepest trench on the planet, in pressure that would fold a steel sphere the wrong way. We have mapped seamounts, sampled brine pools, followed whales into the black. When humans decide to look at something in the ocean, we generally find a way. So distance isn't the barrier. Depth isn't the barrier. Cold isn't the barrier. Those things are expensive, and expensive is not the same as impossible. The barrier is time. Every tool we have for studying a living animal is a tool for capturing a slice of it. A dive lasts hours. A camera drop lasts days. A satellite tag lasts months. Maybe a couple of years before the battery dies, or the animal sheds it. A research program lasts as long as the funding. A scientist's entire working life lasts, if they're lucky, 40 years of fieldwork. Now imagine trying to understand a human being. Birth, childhood, mating, aging, death, using observations that each last about 4 seconds, taken decades apart, from a camera you drop out of a helicopter and never see again. You would learn that humans exist. You would learn roughly what shape they are. You would learn nothing about what they do. That is our relationship with the Greenland shark. Not because it hides, because it is running on a clock that our entire scientific method was never built to read. Every confident sentence you will ever hear about this animal is a still frame, pulled from a dead body on a boat deck, or a few minutes of footage from a camera in black water, or a tag that popped off 2 winters ago and drifted to the surface. We have thousands of still frames. We have almost no film. And that's the reframe. The Greenland shark is not mysterious because it lives somewhere we can't reach. We reach worse places on purpose. It's mysterious because of what it does with its time, and specifically because of how slowly it does everything, which sounds like a weakness, a defect. An animal that never learned to move properly. It isn't. Slowness is the strategy. It's the reason this shark can live where almost nothing its size can. The reason it can afford to be half blind. The reason its flesh is chemically hostile. And the reason it has stayed invisible to us while we stared directly at it. To understand any of the rest of this, you have to understand the slowness first. And the slowness is stranger than it looks. Watch one swim and time the tail. Not the body, the tail. Pick a point on the water and count how long it takes the caudal fin to complete one full sweep, left to right and back. In most large sharks, you can't do this without slow motion. The tail is a blur of correction and thrust, several beats a second, constant. In a Greenland shark, you can count it on your fingers. One stroke. Then a pause that feels like a mistake. Then the next stroke. The recorded figures put the cycle somewhere around 7 seconds. One lazy sweep of a tail the size of a car door, and then nothing, and then another. The whole animal moves at roughly 1 kilometer an hour, somewhere in that neighborhood. That's the cruising speed we've managed to measure, and even when they push, when something is happening, when it matters, the fastest speeds recorded barely double it. Here's what that means in a picture you can actually use. You are walking down a street right now at a comfortable pace. You are moving 4 or 5 times faster than a Greenland shark cruises, drop you in the water, and have you swim badly, arms flailing, no technique. And you would still pull away from it. Not for long, because you'd freeze, but you would win the race. A shark that weighs as much as a great white cannot outswim a tourist doing a bad breaststroke. So the sleeper name fits. Obviously it fits. The animal is broken. Some evolutionary dead end that lost the plot, drifting around the Arctic, waiting to be replaced by something better. That's the read. And it's wrong in a way that's worth taking apart carefully because the mistake isn't in the observation. The observation is fine. The mistake is in assuming that slow is a failure rather than a bill somebody refused to pay. Start with the water it lives in. Not cold, the way you're imagining cold. This is water sitting a degree or two above freezing, and in places brushing below zero without turning to ice, because salt water doesn't play by fresh water's rules. That's the ambient temperature of the shark's entire world. And here is the thing that follows from it. The Greenland shark does not fight it. That's not a small choice. Some sharks do fight it. The great white, the mako, the porbeagle. These animals run a heat exchanger in their bloodstream, a countercurrent system that traps the warmth their muscles generate and keeps their core well above the surrounding sea. Warm muscle contracts faster. Warm nerves fire faster. Warm eyes and brains process faster. That's how a mako becomes the fastest shark in the ocean. It's how a white shark can hit a seal from below hard enough to leave the water. That costs enormously. Being warm in cold water is like heating a house with the windows open. The ocean is stealing your heat every second you exist, and you pay for it in food. Warm-bodied sharks are hungry sharks. They must eat constantly to fund the temperature they've chosen. The Greenland shark opted out. It sits at the temperature of the water around it, 1 or 2 degrees. And when you drop a body to that temperature, the chemistry doesn't just get uncomfortable, it gets slow, in a specific and measurable way. Every biochemical reaction in a cold-blooded animal runs at a rate governed by heat. Chill the tissue, and enzymes work slower. Muscle fibers contract slower. Nerve signals crawl. Oxygen gets consumed at a fraction of the rate. This isn't a Greenland shark quirk. Cool almost any ectotherm and it slows down. The difference is that most ectotherms are visiting the cold. This one lives there permanently. So the 7-second tail stroke may not be a decision at all. It may be the fastest that muscle can physically move at that temperature. The shark isn't choosing to be slow. The water is choosing for it, which sounds like the answer. It's tidy, it's physical, it explains the observation, and it's about half of what's going on. Because if slowness were purely a punishment, a defect imposed by cold, you'd expect the animal to be miserable at it, struggling, barely hanging on at the edge of what's survivable. Instead, it's enormous. It's abundant enough that fishermen kept pulling them up for decades, and it's living in a place where being expensive is a death sentence. Look at what the Arctic deep actually offers a predator this size. No sunlight worth mentioning, no plants, nothing growing. Every calorie down there arrived from somewhere else, sank from above, swam in from elsewhere, or died at the surface and fell. Food is not a resource in that world. It's an event. Something happens, and you're either close enough to reach it or you're not. Now put a warm-blooded predator in there. Give it speed. Give it reflexes. Give it a Mako's burn rate. It's dead. Not because it can't catch anything. It can catch everything. But because it needs to catch something constantly, and constantly isn't on the menu. The furnace runs whether or not there's fuel. In a place where meals are rare and unpredictable, a high metabolism isn't a weapon. It's a countdown. The Greenland shark has almost no countdown. That's what slow buys. Running near freezing with an unhurried tail, this animal's energy demand is a fraction of what a body that size would normally cost. It's not swimming to a meal. It's coasting toward one on a budget so low that finding food occasionally is enough. And that budget gets propped up by something inside it, a liver so large and so loaded with oil that it works as a flotation device. Oil is lighter than seawater, enough of it, and the shark stops sinking. It doesn't have to generate lift with its body, doesn't have to burn energy just to stay off the bottom. It hangs. The most expensive thing about being a fish, not falling, is largely handled. So the picture flips. This isn't an animal that failed to be fast. It's an animal that stripped out every cost it could survive without, and what was left was slow. The slowness isn't the design. It's what the design left behind. And that reframe is worth holding, because it changes what you're looking at. When that gray shape passes the camera, you're not watching a lazy fish. You're watching something running on close to nothing, in a place that offers close to nothing, and staying alive for reasons we're only starting to get at. But there's a hole in this, a big one. And you can see it if you go back to the dead sharks and what was inside them. Everything I just described works beautifully for a scavenger. Cheap body, low demand, no hurry, drifts around, finds a dead thing, eats. It's a coherent animal. It makes sense. It doesn't make sense of the seals. Because look at what a seal actually is underwater. Not on land, where they're comic and slumped and useless. Underwater. In the water, a seal is one of the most capable swimmers in the ocean. It turns inside its own body length. It accelerates like something switched on. Everything about it, every muscle and flipper and instinct, has been shaped by millions of years of running away from things that hunt it. And the things that hunt it are fast: orcas, white sharks, polar bears at the ice edge. A seal's entire existence is a solved problem in escape. Now put the Greenland shark next to it. 1 kilometer an hour, 7-second tail stroke. A body that has surrendered every advantage speed could offer. It's not close. It's not even in the same conversation. The seal isn't slightly faster. The seal is faster the way a car is faster than a person leaning against it. It isn't just seals. Go through the list of what turns up in those stomachs and run the same comparison. Cod, faster. Squid, faster dramatically. Halibut, redfish, smaller sharks, all of them faster. Line up every animal that has ever been found inside a Greenland shark, and you will not find one that couldn't leave it behind without trying. This shark is slower than its own diet, all of it. There are only two ways out of that. Either everything it eats was already dead when it got there, which the condition of some of those remains makes hard to swallow, or the slowest large predator in the ocean has a way of getting close to the fastest prey in the ocean, and we have never once seen it happen. That second option is where this stops being a story about a strange fish and becomes something worse. But before we can go anywhere near it, there's a more basic question sitting underneath all of this, and it's the one that broke everything else open. Not how it hunts, not what it eats. How old is it? Because that question should have had a boring answer. It didn't. Every question about this animal collapses into one question, and it's the one that sounds easiest. How old does it get? That should be a Tuesday afternoon of work. We age animals constantly. It's routine, unglamorous, background science. The sort of thing a graduate student does with a microscope and a lot of coffee. Cut a tree, count the rings. Pull a tooth from a whale, section it, count the layers. Take a scale from a fish, or better yet its otolith, the little stone in its inner ear, and count the bands. Wildlife biology has aging solved. It's a solved problem across half the tree of life. The trick underneath all of it is the same. You need a body part that keeps a record. That's the whole game. Find a tissue that grows steadily, doesn't get remodeled, doesn't get recycled, and just keeps stacking new material on top of old material like sediment on a lake bed. Then read the stack. The reason it works is almost boring. Living things don't grow at a constant rate. They grow faster when it's warm, when there's food, when the season turns. Slower when it's cold and lean. That rhythm gets written into the tissue as alternating layers, dense and sparse, light and dark. And each pair is a year. So you don't measure age. You count seasons the animal lived through. The animal keeps its own diary, and you read it after it's dead. Fine. Do that to a Greenland shark. Here's where it stops. Sharks don't have bones. Not a single one. The entire skeleton is cartilage. And cartilage, in most of the animals you're familiar with, is the soft stuff. Your ears, your nose, the springy padding in your joints. Nothing you could count rings on. Cut a shark open, though, and it isn't quite that simple. Sharks aren't jellyfish. Their cartilage gets reinforced with mineral in the places that need to hold load. And the vertebrae are one of those places. Calcium salts get laid down in the vertebral centra, and they get laid down in bands. Dense in one season, less dense in another, which means for most sharks, you can section a vertebra and read it. Not as cleanly as a tree, and with arguments about what a band actually means, but it works. It's the standard. So you take the Greenland shark's vertebrae, section them, put them under the scope, and there's nothing there. Nothing legible. Not faint. The vertebrae of this animal are so poorly mineralized that the record was never written. There is no dense season and sparse season stacked in alternation, because there isn't enough mineral in the tissue for the pattern to exist. You're looking at a blank page, which, in hindsight, is exactly what you should expect. Go back to the slowness. This is an animal that stripped out every metabolic cost it could survive without. Mineralizing a skeleton is a cost. Hauling that mineral around for your entire life is a cost. It makes you heavier, and heavier means burning energy to stay off the bottom, which is the one thing that giant oil-filled liver exists to prevent. A Greenland shark that built a properly reinforced skeleton would be paying twice for a thing it doesn't need. It's not fighting currents. It's not accelerating. It's not slamming into prey at speed. Its skeleton has to do almost nothing. So it doesn't. Look at what that means, though. The same austerity that lets this shark live where it lives is the reason it doesn't keep a diary. The cheapness is the point, and the cheapness erased the record. The animal is not hiding its age from us. It simply never bothered to write it down because writing it down would have cost energy it didn't have. That's the first trap. Now here's the second one, and it's worse because it looks like a way out. If you can't count layers, measure length, catch a bunch of sharks, tag them, release them, catch them again later, see how much they grew. Growth rate plus size equals age. Crude, but real. It's been done with plenty of species. Try it here. The growth increments that have come out of tagging work on this animal are small, very small. So small that the measurement starts fighting the instrument. You are trying to detect a change that's barely larger than the error in how you measured the shark in the first place, and you'd need to hold the tag on a wild animal in Arctic water for years to get a difference worth trusting. But push through it anyway, do the arithmetic, and you get an answer that nobody wanted. Because if the growth rate is that small, and the animals get that big, the math doesn't produce a shark that's 40. It produces something absurd. Something you'd assume was an error in your arithmetic, because the alternative is that a fish outlives the study, the scientist, and the institution. So the problem sat there. Not a small problem. This is the foundation everything else rests on. You cannot say whether the species is in trouble if you don't know how fast it replaces itself. You cannot say whether it's rare or just slow. You cannot say what its life even looks like. Every serious question about the Greenland shark was locked behind a number nobody could get. For decades, that was the situation. The largest predatory shark in the Arctic. And we couldn't tell you if the one on the deck was a teenager or an antique. Then somebody had an idea that sounds insane until you understand why it works. Forget the skeleton. Look at the eye. Specifically, the lens. And to see why that's not madness, you need to know what a lens actually is. A lens has to be transparent, which is a genuinely difficult thing for living tissue to be because the machinery of a living cell, the little organelles, the structures, the churn, scatters light. So the lens does something drastic. Its cells strip themselves out. They lose their internal machinery and fill up with tightly packed protein, arranged so precisely that light passes straight through, which makes them clear and also makes them dead, functionally. There's no blood supply in there, no turnover, no repair crew. The proteins at the very center of the lens, the nucleus, get laid down while the animal is still developing before it's born, and then they are never replaced, ever. They sit there for the entire life of the animal, exactly as they were made. Read that again because it's the whole thing. Every other tissue in the body cycles. Your muscle isn't the muscle you had years ago. The atoms get swapped. The proteins get broken down and rebuilt. A body is a process, not an object. But the center of the eye lens is frozen the moment it forms. It is physically a piece of the embryo, preserved intact inside an adult, carried around for as long as that animal lives. It's a time capsule. It was never meant to be one. It's a side effect of needing to see, and the shark has been carrying it the entire time. So the picture reverses. This is an animal that refused to record its own age anywhere. No bones, no bands, no rings, a skeleton deliberately too cheap to keep notes. And the one archive it couldn't avoid keeping is the piece of tissue it needed to be transparent. The record survived because it was clear. Now hold on to the other half of this, because it's grim. And it comes back. The eye. Of all the organs on this animal, the one carrying the only readable history is the eye. And if you've seen a photograph of a Greenland shark's face, you know something is wrong with it. There's a thing hanging off the cornea, a pale worm-like shape, drifting in the current, attached to the front of the eyeball. The one clock this animal has, and something is sitting on it, eating. That's for later, because there's still a gap here, and it's the size of a canyon. You have a piece of tissue that formed before birth and never changed. Great. It doesn't have rings. It doesn't have layers. It's just a ball of ancient protein with no seasons written in it. Knowing it's old doesn't tell you how old. You need a way to ask a lump of protein when it was made, and nothing in biology does that. Something else does. Something that has nothing to do with sharks or oceans or the Arctic at all, and everything to do with what humans were doing to the atmosphere in the middle of the 20th century. When they finally got the answer out of that lens, it was so far outside expectation that the first reasonable response was to assume the method was broken. 1952, 1954, 1961, 1962. Above the Pacific, above the Arctic, above the deserts of Nevada and the steppe of Kazakhstan, humans set off hundreds of nuclear weapons in the open air. It was the single dumbest chapter of the 20th century, and it is not what you'd call a scientific instrument. It became one anyway. When those weapons went off, they threw neutrons into the atmosphere, and those neutrons hit nitrogen, and nitrogen turned into carbon-14. The same radioactive carbon that already existed up there in trace amounts, made continuously by cosmic rays. Except now it wasn't a trace. Within about a decade, the amount of carbon-14 in the atmosphere roughly doubled, a vertical line in the global chemistry of the air, sharp as a knife edge, and then a slow decline afterward as the oceans and the biosphere soaked it up. That spike is called the bomb pulse. And here's what makes it useful. Carbon doesn't stay in the air. It dissolves into the surface of the ocean. It gets fixed by plankton. Plankton get eaten. That elevated signal moved down through the marine food web with a lag of a few years, and it went into everything that was building a body out of the ocean's carbon at the time. Everything alive after the early 60s got stamped. Everything alive before it didn't. The world accidentally date-stamped itself. And biologists, being opportunists, started using it. Fish otoliths, coral, sediment, anywhere you have inert tissue that formed once and never turned over, you can look for the spike. If it's there, the tissue formed after the bombs. If it's absent, before. Now put that together with the eye. The nucleus of the lens forms before birth and never changes. It is made of carbon that came out of the ocean at the moment the animal was developing. So drill into the center of a Greenland shark's lens, pull out that ancient protein, run it through a mass spectrometer, and you are asking a very specific question. What was the carbon of the North Atlantic like on the day this shark was made? They did it on sharks that came up as bycatch, animals already dead, across the full range of sizes, from small ones to the giants. The small sharks lit up. The bomb pulse was there in the lens nucleus, elevated carbon-14 clear as a signature. Those animals were born after the tests. Fine. Expected. That's the method working. Then they went to the big ones. Nothing. No spike. The lenses of the large Greenland sharks carried carbon from a world that had never heard a bomb go off, which is the first result, and it already breaks something, because the bomb pulse is a floor. It doesn't tell you how old those sharks are. It only tells you they cleared the line. Every large shark in that sample was born before the early 1960s, and by that point, the world had cars and antibiotics and 2 world wars behind it. You need the second half of the technique to go further, and the second half is older and less forgiving. Carbon-14 decays, slowly. Half of it is gone after about 5,700 years. And that slowness is the problem. Over a few centuries, the amount that disappears is small. You are trying to measure a shallow slope on a curve that was built for measuring things thousands of years old. Then you have to correct for the fact that ocean carbon isn't the same age as atmospheric carbon, because the deep sea holds carbon for a long time before it comes back up, and that offset varies by place and depth and era. So the answers don't come out as numbers. They come out as ranges, wide ones. And this is the part where you have to decide whether you want the truth or the headline, because the headline number is famous and the truth is more interesting. The central estimate for the sharks in that sample landed around 272 years, not the maximum. The middle of the distribution. The typical large Greenland shark in the sample was, on the best available reading, close to 3 centuries old. The largest individual returned an estimate near 392 years, with an uncertainty of roughly 120 years on either side, which means the honest way to say it is that this animal was somewhere between about 272 and 512 years old, and the method cannot narrow it further. Look at what that range actually contains. At the low end, that shark was born around the time Isaac Newton was alive. At the high end, it was already swimming before the Mayflower left England. That is not a small margin of error. That margin of error is longer than the entire history of the United States, and it sits inside the life of one fish. The uncertainty is the finding. People who quote a single number are flattening the strangest part of this into a trivia fact, because here's what a range that wide tells you. It tells you we are not measuring this animal. We are bracketing it. We have a technique that can say several centuries and cannot say which several. We have the longest-lived vertebrate known to science, and we cannot date it as precisely as an archaeologist can date a piece of charcoal. Now stack the twists, because they get worse. That shark was not the oldest Greenland shark. It was the oldest one that happened to die in a net that somebody happened to sample. There is no reason to think the sampling caught the ceiling. If anything, the ceiling is almost certainly higher. You don't stumble onto the record holder by accident, you stumble onto whatever was nearby. Every maximum age you have ever heard for this species is a floor wearing a crown. The method has critics. It should. It rests on assumptions about exactly when the lens nucleus locks in, about how carbon from the surrounding ocean gets incorporated, about which calibration curve applies to Arctic water at depth. Push on any of those, and the numbers move. Some researchers have pushed. That argument is not settled, and anyone who tells you it is has stopped reading. And here's what makes the argument almost impossible to end. Normally, you validate a dating method against something you already know. You take an animal of confirmed age, tagged young, recovered old, and you check whether the technique returns the right answer. That's how radiocarbon earned trust for other fish. You test it on a case where the truth is already in hand. You cannot do that here. To validate a 300-year clock, you would need a shark somebody tagged 300 years ago. Nobody did. Nobody could have. The method that finally gave us a number is a method we have no independent way to confirm, and the only experiment that would confirm it would have had to start before the technique was invented. So the number stands on its own internal logic. That's not nothing. It's also not the same thing as proof. The debate doesn't rescue the old picture, though. That's the thing people miss. Nobody arguing about the calibration is arguing for a 40-year-old shark. The disagreement is over whether the answer is 2 centuries or 5. The floor moved, and it never moved back. So the Greenland shark is, on the best evidence we have, the longest-lived vertebrate on the planet. Probably. With error bars, you could park a boat in. And this is the point where the whole animal reorganizes around one fact, and everything before it stops being a separate observation. The slowness wasn't a defect. You knew that. Now look at what it bought. That refusal to be warm. That refusal to mineralize a skeleton. That 7-second tail stroke. That liver full of oil doing the work gravity would otherwise charge for. Every one of those is a bill this animal declined to pay. And the sum of all those unpaid bills is time. Not speed. Not power. Time. The metabolism is the currency. It always was. Everything the Greenland shark gave up, it converted into duration, which means the thing swimming past the ROV light is not a large fish. It's a long one, long in a direction the camera can't see. That gray shape has been moving through that water since before the country you live in had a name. Since before the language you're hearing this in sounded like it does now. And it has never once been in a hurry. Because hurry is what it traded away to be there at all. That should feel like an ending. A clean one. The mystery of the slow shark solved. Slow equals cheap equals long. It isn't an ending. It's the beginning of a much uglier problem. And it comes from a question you'd only ask if you'd spent time around actual animals. If it takes that long to live, how long does it take to grow up? Nobody had checked. When they finally ran the numbers, the answer turned this species from a marvel into something breakable. Every animal you know has a childhood you could sit through. That's not a poetic claim, it's a scheduling fact. A mouse is breeding within weeks. A cod is a few years. A white shark takes a decade or so, maybe more for females, and that already counts as slow. Slow enough that it's the first thing conservation biologists mention when they explain why shark populations don't bounce back. An elephant needs over a decade. A human takes, 13, 14 years to become physically capable of it. The absolute outliers on land. The big tortoises, the long-lived birds. Stretch it to a few decades and that's the far edge of the map. You can watch any of these happen. That's the point. A researcher can start a study on a young animal and still be alive, still be employed, when that animal has offspring. The whole of reproductive biology is built on the assumption that a career is longer than a childhood. Now do the arithmetic on the shark. Start with what a mature female looks like. The size at which they appear to become sexually mature is enormous. Somewhere in the range of 4 meters based on examining the reproductive organs of animals that came up dead. Below that, the equipment isn't ready. Above it, it is. That's a physical threshold you can see. 4 meters. Put that against something real. That's longer than a compact car. It's roughly the length of a small skiff. A boat 2 people would work from. So the shark has to grow to the size of the boat that catches it before it can reproduce once. Then take the growth rate. Remember the tagging problem from earlier. The increments so small they fight the ruler. The best figures we have suggest a Greenland shark adds around a centimeter a year, roughly. Call it the width of a fingernail. Now divide. You are asking how long it takes to build 4 meters of animal at the rate of a fingernail per year, in water a degree above freezing, on a metabolism that was designed from the ground up to consume as little as physically possible. The number that came out of that work was around 150 years to reach maturity. With uncertainty on it, obviously, it's built on a growth rate we can barely measure and an age technique with error bars you could park a boat in. Some of the estimates run lower, some run higher. Nobody would defend the precise figure. But the shape of the answer doesn't move. Whatever the exact number, the Greenland shark spends more than a century as a juvenile. Sit with what that sentence contains. There is a shark in the water right now 3 meters long, weighing more than you, with a full set of teeth and a functioning brain and a stomach that has been digesting things for 100 years, and it is a child. Biologically, it has never reproduced. It cannot. Its entire existence so far has been growth, and it has decades left before the equipment comes online. An animal that has been alive since the invention of the telephone is not yet an adult. Here's the immediate consequence, the one that turns this from a curiosity into something with teeth. No human has ever watched a Greenland shark grow up, not part of it. I mean the whole arc. Birth to first reproduction, the fundamental unit of a species' existence. It cannot be done. It is not a funding problem or a technology problem. You would need a research program that outlives the researcher by a century, then hands off, then does it again, and the thing you would be watching for the entire time is a fish getting 1 centimeter longer per year. Every study of this animal is a person looking at a slice of a life they will never see the ends of. We are not slow at studying it. We are structurally incapable of studying it. That's not the same problem. Which brings us to the reversal, and it's the one that matters most in this entire story. Long life sounds like an advantage. That's the instinct, and it's almost impossible to shake. Living for centuries feels like winning. It feels like this animal solved something the rest of us didn't, that it found the exit from the problem everything else is stuck in. Turn it over. Look at the underside. A species doesn't survive by having individuals that live a long time. A species survives by replacing itself. That's the only metric that has ever mattered. Longevity is not the goal. Longevity is a strategy for reaching the goal, and it only works under one condition: nothing kills you. That's the deal the Greenland shark signed. In the Arctic deep before humans, that was a reasonable bet. What's going to kill a 4-meter shark down there? Nothing hunts it at that size. Nothing competes with it seriously. The environment is stable in a way surface ecosystems never are. No storms, no seasons that matter, no droughts, no predators. So an animal can afford to take 150 years getting to adulthood, because in that world, the odds of dying before you get there are genuinely low. Take your time. Nothing's coming. The strategy is not live long. The strategy is die rarely. Longevity is the symptom, and a strategy built on nothing killing you is the single most fragile thing in biology, because it has exactly one failure mode. Something starts killing you. Now run a mouse through the same test. A mouse breeds in weeks and dies in a year or two, and that looks pathetic until you realize it's armored against catastrophe. Wipe out 90% of a mouse population and the survivors rebuild it in a season. The rate of replacement is so fast that mortality barely registers. You cannot kill mice faster than mice make mice. That's why they're everywhere. The Greenland shark is the exact inverse. Every individual that dies takes a century and a half of construction with it, and the replacement has to start from zero and run the whole clock again. There is no fast rebuild. There is no compensating burst. The species has no gear for it. So the population sitting in the North Atlantic right now is not a population in the normal sense. It's an inventory. Standing stock that took centuries to accumulate, and every single one you remove is subtracted permanently on any timescale a human would recognize. Here's the worst part. Go back to that liver fishery. Decades of it. Thousands of animals hauled up, killed, cut open, the oil taken. Every one of those sharks was somewhere on the growth curve. Some were adults, and a great many, probably most, given how the size distribution works, had not reached maturity yet. That means we killed animals that had been alive for 100 years and had never reproduced. Not once. An entire century of existence, of surviving the cold and the dark and the scarcity, of getting to the size where it was almost ready. And then a line came down, and that was it. The genetic contribution of that individual to its species. 0. It was a century of biological investment, canceled before it paid out. We were not harvesting a resource. We were harvesting the future of a species. And we couldn't tell the difference because a 100-year-old juvenile and a 200-year-old adult look almost exactly the same on a deck. You cannot see maturity. It's not written on the outside. That's the reframe. And it's ugly. The longevity of the Greenland shark is not a superpower. It's a debt. It's a species that spent everything on duration and left nothing in reserve for the possibility that something might start removing it faster than it can rebuild. And then we showed up, and we removed it faster than it could rebuild. without ever knowing what we were doing because nobody had run the numbers until decades after the fishery was over. The animal that beat time cannot survive being in a hurry. But there's a hole in everything I just told you, and once you see it, you'll understand why this species is so much stranger than the age number makes it sound. I said they mature around 150 years. I said they reproduce after that. I described the whole arc: birth, growth, maturity, offspring, like it's an established sequence. It isn't. Every part of that comes from cutting open dead animals and inferring what the organs were for. Nobody has ever seen a Greenland shark mate. Nobody has ever found a pregnant one and watched what happened next. Nobody knows where they give birth, or when, or how many, or where the young ones spend their first decades. We have thousands of dead adults and almost no beginnings. The most important event in the life of the longest-lived vertebrate on Earth is completely, absolutely invisible to us. Here's a fact that should not be possible in the 21st century. We have sequenced this animal's genome. We have read its DNA. Billions of letters, the full instruction set, laid out and searchable. We can tell you things about its molecular architecture that no fisherman in 300 years could have guessed. We have never seen two of them mate. Not once. Not a glimpse, not a partial, not a blurry frame from a drop camera that somebody argued about for a decade. Zero. The most fundamental act in the life of any sexually reproducing animal, the thing the entire body exists to accomplish, and for the longest-lived vertebrate on Earth, it is a complete blank. We know its code. We don't know how it makes another one. So what do we actually have? Let's be honest about the evidence, because the honesty is the interesting part. We have dead females. A small number of them, and I want you to hold on to how small, that came up carrying young. From that, we can say the species is not laying eggs on the seafloor. The young develop inside the mother, in the way most sharks do it: eggs that hatch internally, embryos that grow on yolk rather than a placenta, and then live pups delivered into the water fully formed. That's the mechanism. That much is real. Everything else you have ever heard about Greenland shark reproduction is an extrapolation from a handful of specimens. The litter size number gets quoted a lot, around 10 pups. It appears in summaries, in fact sheets, in every popular article. Trace it back, and you find it resting on a very thin foundation. A small number of historical records of pregnant females, examined on decks by people who were not there to do reproductive biology. That is the entire empirical basis for what we say about how many offspring this species produces. Compare that to what we know about a cod, or a herring, or a great white. Those animals have been studied with survey after survey, hundreds of individuals, statistical distributions. For the Greenland shark, we have a number that essentially comes down to somebody cut one open, and this is what was inside. It might be right. It might be badly wrong. Nobody can tell you, and anyone who quotes it without that caveat is passing along a rumor with a decimal point. Now the gestation. How long is a Greenland shark pregnant? I don't know, neither does anyone else. And the reason this question is genuinely maddening rather than just unanswered is that we have no idea what the sensible guess even looks like. Think about how you'd normally estimate it. You'd look at related species and scale. Big shark, long pregnancy. A basking shark's gestation is thought to run for years, and that already sits at the outer edge of vertebrate biology. So you'd say, Greenland shark, similar size, maybe similar. A few years, reasonable. But nothing else about this animal follows the normal scaling. It grows at a centimeter a year in water at 1 degree. Its entire metabolism has been dialed down to the point where it takes 15 decades to reach adulthood. If embryonic development runs on the same slowed-down chemistry as everything else in that body, and there's no obvious reason it wouldn't, then the pregnancy could be far longer than anything we have a category for. Or it might not be. The embryos might be running on yolk in a warmer part of the mother's body, or the mother might move to different water while carrying them, or development might be buffered in some way we've never thought to look for. We have no observations. We have no framework. The honest answer is that the gestation of the Greenland shark could plausibly be measured in years or in decades, and we cannot currently distinguish between those two possibilities. Read that again. We are unable to say whether a pregnancy in this species lasts closer to 2 years or 20. That's not a gap in the literature. That's a hole where the literature should be. It gets stranger when you go looking for the pups. Newborn Greenland sharks are small. The smallest recorded individuals are around 40 centimeters, which is nothing. Arm's length. That's the starting size of an animal that will end up 4 or 5 meters long and still be growing. Now, where are they? Because in every other shark species we study, this is a question with an answer. Nursery grounds are one of the standard findings of shark biology. Sandbar sharks use estuaries. Lemon sharks use mangrove flats. Great whites have coastal nursery zones where the juveniles congregate before they move offshore. Find the nursery, and you've found the future of the population. It's where conservation actually happens, because that's where the young ones are concentrated and vulnerable. For the Greenland shark, there is no known nursery anywhere in the entire North Atlantic and Arctic. We have hauled up thousands of these animals over a century of fishing. Halibut longlines catch them constantly as bycatch, all across their range. At every depth, people fish. And the catch is dominated by large sharks. The small ones, the 40-centimeter pups, the 1-meter juveniles, the whole first stretch of the growth curve, barely appear. 2 ways to read that. Maybe they're there, and we're not catching them. Gear selectivity is real. A hook baited for a large shark doesn't reliably catch a small one. That would explain the absence without requiring anything strange. Or maybe they're somewhere else. Somewhere we don't fish, don't survey, don't look. Somewhere deeper or further under the ice, or in a part of the water column nobody has bothered to sample because there was never a reason to. If that's true, then there is an entire life stage of the largest Arctic shark, possibly the first 50 years of its existence, happening in a place we have never located. 50 years of an animal's life, and we don't know what continent it's near. That is a genuinely absurd sentence to write about a vertebrate in this century. And it doesn't feel absurd when you're inside the problem, which is exactly the trap. Every individual fact is boring. Bycatch skews large. Nurseries are hard to find. Deep water is expensive. Nothing on that list is shocking. It's only when you step back that you see the shape. We have never located the place where this species begins. So here's what the reproductive biology of the Greenland shark actually consists of. We think mating involves the male biting the female to hold position, because that's what sharks do. And there are reports of scarring on females that would be consistent with it. Inference from relatives, not observation. We think it happens somewhere deep, because that's where the adults are. Inference from distribution. We think the pups are born live and fully formed, based on a small number of dead pregnant females. We think litter size is around 10, based on almost nothing. We think maturity comes at around 150 years, based on a growth rate we can barely measure, and an age technique with wide error bars. We do not know when. We do not know where. We do not know how long. We do not know how often. Whether a female reproduces every few years or once a decade, or a handful of times across 3 centuries. That last one matters more than any of the others, and it's the one nobody can touch. Because if a female Greenland shark reproduces rarely, if the total lifetime output of a 300-year-old animal is a few dozen pups spread across a century and a half of adulthood, then the arithmetic of this species is far worse than the maturity number alone suggested. A population like that doesn't recover from losses. It doesn't recover from anything. It just gets smaller, and we cannot even check. Which brings us back to the thing this whole story keeps circling. Not the cold, not the depth, not the remoteness. Every single one of these blanks has the same cause underneath it, and it's the one from the beginning. Everything we know came from dead sharks. And a dead shark cannot show you a verb. You can cut open 10,000 of them, and you will never see one mate. You will never see one give birth. You will never see one grow. Those are things that happen in time, and time is the one dimension a corpse doesn't have. So we sequenced the genome instead, because that's what you do when you can't watch. You take what the body will give you, and you read it in a lab, where the animal's timescale doesn't apply. We know the letters. We don't know the sentence. And the eye, the one organ that finally cracked the age problem, the one piece of tissue that kept a record when the rest of the body refused to. That eye has something living on it, something that has been there in the photographs the whole time. Look at a photograph of a Greenland shark's face, not a diagram, a real photograph, taken on a boat deck or through a camera housing in black water. There's something hanging off the eye. It's pale, 2 or 3 centimeters of soft body anchored to the front of the eyeball, with a pair of long threads trailing behind it into the water. It doesn't look like an injury. It looks like a piece of equipment somebody bolted on. And once you've seen it, you can't look at anything else on the animal's face. That thing is alive. It's a copepod, Omatocoida elongata. And copepods are crustaceans. Same broad family as crabs and shrimp. Most of them are microscopic, the drifting confetti of the plankton, the thing that baleen whales strain out of the ocean by the ton. They are food. They are the bottom of the food chain, the definition of it. One of them is eating the eye of a 4-meter shark. The anchoring end burrows into the cornea and holds. The body hangs out into the current. The trailing threads are egg strings. The animal is reproducing right there, on the surface of the eyeball, releasing larvae into the water to go find another shark's face. The cornea is not a way station for this species. It is the habitat. Its entire adult existence is spent attached to the front of a Greenland shark's eye, feeding on the tissue underneath it. And it does damage. Real damage. Corneal lesions, scarring, the transparent surface of the eye chewed into something that is no longer transparent. In heavily affected animals, the eye is not a functioning optical instrument anymore. It's a wound with a crustacean living in it. It isn't a small tenant on a large host. Hold one next to a human eye, and it would cover most of the iris. This is a parasite you could lift off with 2 fingers, riding the face of an animal the size of a car, and the animal has no answer to it at all. Now here's the number that turns this from grotesque into a genuine biological problem. This isn't rare. In sample populations, the great majority of Greenland sharks carry these parasites. Not a minority, not the sick ones. Most of them, in some samples, the overwhelming majority, and frequently on both eyes at once. Whatever this animal is, being half-blind from a crustacean is not a misfortune that befalls the unlucky ones. It is the standard condition of the species. So the largest shark in the Arctic swims around with parasites eating its eyes, and it apparently does not care. Stop there, because that should be impossible. Take any predator you can name and start blinding it. A great white, a lion, a hawk. You have just ended that animal. Vision is the primary hunting sense across most of the vertebrate world. It's how you find prey, how you time the strike, how you judge distance in the last half second when everything is decided. Damage the eyes of a predator, and you have not inconvenienced it. You have removed its job. Evolution does not tolerate that. If a parasite were destroying the hunting capacity of a species, that species would be selected into a response. A defense, a behavior, a physical barrier, something, or it would be selected out of existence. What you do not get is an animal that shrugs. The Greenland shark shrugs, which means one of two things is true. Either the shark can't do anything about it, or it doesn't need to. Take the first one, can't. And there's a real case here because look at what this animal has to work with. It has no hands. It has no cleaning station. The reef fish that pick parasites off larger fish are a warm water arrangement, and there's nothing like that in the Arctic deep. It can't scrape the parasite off, because scraping requires acceleration and precision against a hard surface, and this is an animal that moves at 1 kilometer an hour and takes 7 seconds to complete a tail stroke. You cannot rub your eye against the seafloor when it takes you a minute to turn around. It can't outrun the larvae either. They're in the water. They're drifting. The shark moves so slowly through so much of it for so long that the encounter is essentially guaranteed. Given 3 centuries in that water column, you will meet everything the water column has to offer. That's the first twist, and it's a good one. The parasite load might not be a disease. It might be a receipt. Centuries of exposure accumulated on a body that had no mechanism to refuse. The reason the eyes are ruined is not that this shark is uniquely vulnerable, it's that it has been alive long enough for the unlikely to become inevitable. The infestation is a symptom of the longevity, not a threat to it. But that only explains why the parasite arrives. It doesn't explain why the shark is still functional. Which brings us to the second option, and this is the one that reorganizes the animal. The shark doesn't need to do anything about it, because the eye was never worth much. Go back to where this thing lives. Deep Arctic water, often under ice, hundreds of meters down and frequently much further. Sunlight does not reach that, not dimly. At the depths this animal spends its time, the sun is a rumor. There is no image to form. There is nothing for a cornea to focus, because there is no light going through it. What exactly is the parasite destroying? An organ that was already sitting idle. Vision in that environment isn't the primary sense. It might not be a meaningful sense at all. The thing on the eye is eating a piece of hardware the shark had already stopped depending on. And that's why nothing in 300 years has pushed back against it. Evolution isn't tolerating an injury. It's declining to pay for a repair on something it doesn't use. Look at how cleanly that fits everything else. This is the animal that refused to be warm, that refused to mineralize its skeleton, that refused to fund a fast tail. It is the same accountant every time, making the same decision. If it doesn't earn, it goes. Defending the eyes would cost energy, an immune response, a behavior, an anatomical structure, and the eyes weren't earning. So there's no defense. Of course there's no defense. There was never going to be one. The cheapness explains the parasite the same way it explained the blank vertebrae. Now you've probably heard the other version of this story, because it's the version that spreads, the one where the copepod glows, where it hangs off the eye, emitting light, and pulls curious prey toward the shark's mouth in the darkness. And the whole thing is a beautiful, accidental partnership. The parasite pays rent by fishing for its host. I want to be direct about this. That idea is not established. It has been raised, it circulates constantly, and it is enormously appealing. It is also not supported by good evidence. There is no solid demonstration that this copepod produces light, and no observation of prey being drawn to it. It is a hypothesis that became a fact by repetition, which is a different thing from becoming a fact by evidence. Notice why it spreads. It's satisfying. It converts a horror into a bargain. It makes the parasite useful, which makes the shark clever, which makes the whole arrangement feel designed. We prefer stories where the disgusting thing turns out to have a purpose. The evidence points somewhere less comfortable. There's no bargain here. The shark isn't being helped. It's being fed on, in the eye for centuries, by a relative of plankton. And it survives because the organ under attack was never critical enough to defend. That is a stranger animal than the one with a glowing lure. It's an animal whose eyes are so unimportant that a crustacean can farm them without consequence. So the picture inverts one more time. You look at that photograph and you see a wound, a victim, something wrong. What you're actually looking at is a piece of anatomy the shark has effectively abandoned, hosting a tenant that took over an empty building. And remember what the eye is. Remember the lens, the one archive this animal keeps, the only tissue that recorded when it was born, the single piece of it that could answer the question everything else depends on, the one clock this species has. And something is sitting on it, eating. We got our answer out of that eye, barely. And the animal itself got nothing from it because it stopped using it long ago. But that leaves the hole. That leaves the thing we've been walking toward without saying. If the eyes are junk, if the largest predator in the Arctic is functionally blind and doesn't care, then it is finding food in total darkness, in a place where food is rare, moving at 1 kilometer an hour. With what? Turn the lights off in your house right now and find the fridge. You can do it. You'd bump a door frame, but you'd get there because you've built a map of that space over years and you're navigating memory, not information. Now do the same thing in a stranger's house, in a house the size of a country, where the fridge moves. That's the actual problem, not darkness. Darkness is trivial. The problem is a volume of water measured in cubic kilometers, containing almost nothing. And somewhere in it, occasionally, something edible. Sunlight is not a factor here. Not dimmed, not filtered. The top of the ocean absorbs it so thoroughly that by the depths where this shark spends most of its life, the sun has simply stopped being a thing that exists. And what light does occur down there isn't sunlight at all. It's biological. Little flashes made by animals, brief and local, which are useful for signaling and ambushing, and not much use for finding a body on the seafloor 2 kilometers away, so the eye is out. We established that, and the shark stopped defending it centuries ago for exactly this reason. What's left is chemistry, pressure, and electricity. Those are the 3 things that still work when photons don't. And every deep-sea predator on Earth is built out of some combination of them. Start with smell, because for sharks, that's the famous one. And the famous version is wrong in a way that matters. You've heard the claim, a shark can smell 1 drop of blood in an ocean. It's nonsense as stated, and the real thing is more interesting anyway. What a shark's nose actually is, structurally, is a pair of chambers under the snout with water flowing continuously through them as the animal moves forward. Not connected to the throat. Sharks don't breathe through their noses. The nostrils are pure sampling equipment. Water in one side, across a folded sheet of sensory tissue out the other. A chemical detector with a permanent current running through it. The sensitivity is real. Sharks can detect certain compounds at concentrations that are difficult to state without sounding like exaggeration, and that's genuine tested biology. But here's the part everyone skips. Sensitivity is not location. Detecting a molecule tells you a thing exists. It does not tell you where. That's a completely different problem, and it's the one that actually has to be solved. A scent in the ocean doesn't radiate outward in a neat expanding sphere with the source at the center. It gets pulled into a plume by the current, a long, ragged, twisting ribbon of chemistry stretching down current from whatever produced it, broken up by turbulence into patches with gaps between them. Concentration doesn't rise smoothly as you approach. You hit a patch, then nothing, then a patch. So a shark following a smell is not walking up a gradient. It's doing something more like a search algorithm. Detect, orient into the current, hold that heading, lose the scent, Cast around until you find it again. Reorient. Continue. The nose says yes. The current says that way. The rest is patience. And this is where the animal we've been assembling walks into the room and everything fits. Because that strategy, hold a heading, don't panic, keep going, expect to lose the signal repeatedly, cover distance for hours without any confirmation you're right, is nearly impossible for a fast animal. Speed burns fuel. A mako doing that search is spending calories at a catastrophic rate on a bet that might not pay out. It cannot afford to be wrong for 6 hours. The Greenland shark can be wrong for a week. That's the reframe. And it's the best one in this whole story. Slowness isn't a handicap in scent tracking. It's the enabling condition. A body running on almost nothing can treat an 8-hour transit toward a maybe as a rounding error. It doesn't need to be sure. It doesn't need the odds to be good. It just needs to be right occasionally. And it has 3 centuries to be right occasionally. The nose finds food, but the metabolism is what makes following it survivable. Now the second system, and this one has no equivalent in your body at all. Run a finger down the side of any fish, and you'll feel a faint line, snout to tail. That's the lateral line, and under it is a canal full of fluid, connected to the water through pores, with sensory cells inside that detect the fluid shifting. What it registers is water movement. Pressure changes. The push and pull of something displacing water nearby. It is functionally hearing with your skin. It is how a school of fish turns as one unit without a signal. How a blind cavefish navigates a wall it's never touched. And how a predator in black water knows that something just moved. Range is limited. This is a close quarters sense. Meters, not kilometers. But it's fast. And it's directional. And it doesn't care about light. In the final approach, when the smell has brought you into the neighborhood, and there's still nothing to see, the lateral line is the thing that tells you where. Then, the last one. The strangest. Look at the front of a shark's snout under magnification, and you'll find pores, hundreds of them, scattered across the skin like someone pressed a needle in over and over. Under each is a tube, filled with a conductive jelly, running back to a bulb of sensory cells. These are the ampullae of Lorenzini, and what they detect is voltage. Not metaphorically, actual electrical fields at strengths that are absurd. Sharks are sensitive to gradients so faint they're measured in billionths of a volt per centimeter, which makes this arguably the most sensitive electrical detection system in the animal kingdom. Here's why it matters. Every living animal leaks electricity. A muscle contracting generates a field. A heart beating generates a field. Even the difference between the salts inside a body and the salts in seawater generates a field across the gills, across the skin, constantly. Whether the animal is moving or not, you cannot turn it off. You cannot hide from it. Holding still doesn't help. A motionless fish buried under sediment is still broadcasting because being alive is an electrical event. So the shark's face is a sensor array reading the electrical noise of everything nearby that has a pulse. In total darkness, against an animal doing everything possible to be undetectable, the ampullae still see a heartbeat. But it's short range. That's the constraint, and it's not a small one. These fields fall off steeply with distance. This is a sense that works across a meter or so, not across a room. It's not a search tool, it's the last step, which is what makes the whole thing click into a sequence. Smell operates at kilometers. It's the long-range detector. It says something is out there. In that direction, go. The shark commits, holds the current, spends hours crossing black water toward a chemical maybe. The lateral line takes over in the last stretch, reading the disturbance of a body moving nearby, narrowing the approach. And in the final meter, when contact is imminent and there is still nothing to see. The ampullae light up the target from its own electricity. 3 systems, 3 ranges, nested inside each other. No light involved anywhere. That's a functioning predator. It's a completely alien one. Nothing about it resembles how you experience the world. But there's no gap in it. The animal isn't stumbling around hoping to bump into dinner. It's running a layered detection stack that starts at kilometers and ends at centimeters. And the ruined eyes are irrelevant to every stage of it. 2 things you should be uncomfortable about, though. The first is honesty. Almost everything I just described comes from shark biology generally, from species we can catch, keep, test, and run controlled experiments on. The specific sensory performance of a Greenland shark, in its own water, at its own temperature, has barely been examined. It has the anatomy, whether the cold degrades it, whether the proportions are unusual, whether it emphasizes one channel over another. We're inferring from relatives, and that's a weaker claim than it sounds. The second is worse. Everything I just built explains a scavenger perfectly. Smell a carcass from kilometers away, take 9 hours getting there, arrive, eat. Every piece of that system is optimized for finding something dead and being in no rush about it. Now put a seal in it. A seal is not a scent plume. A seal is awake. It has ears, eyes, a lateral system of its own, and a nervous system tuned by millions of years of being hunted by things that are fast. It doesn't sit still. It moves at speeds this shark cannot conceive of. The sensory stack tells you the shark can find one. That was never the question. The question is what happens next, because finding a seal and catching a seal are separated by the single largest speed gap between predator and prey in the entire ocean. And the seals are in the stomachs. A seal is in the stomach. That's not a theory. That's a thing you can hold in your hands on a boat deck. And every single thing we've established says it can't be. Line it up. The shark cruises around 1 kilometer an hour with a tail stroke you can count out loud. A seal underwater is one of the most capable swimmers in the ocean. It turns inside its own body length, accelerates like a switch flipping, and has spent millions of years being shaped by things that hunt it at speed. Orcas, white sharks. The gap isn't close. The seal is faster the way a car is faster than someone leaning on it. So how does the slowest large predator in the ocean end up with the fastest prey inside it? The easy answer is scavenging. It's dead already. The shark smells it, takes 9 hours getting there, eats. Everything about the animal supports this. The nose, the metabolism, the patience. It's clean. Except the remains don't always cooperate. Some of what comes out of these stomachs is exactly what you'd expect from a scavenger. Chunks, fragments, the sort of thing you'd find on something that arrived late to a carcass. But there are also reports of seal remains that look wrong for that story. Too intact, too complete. Portions of animals that read less like the aftermath of a fall and more like something that was taken. Here's a detail that quietly does a lot of work. The stomachs contain land mammals, reindeer, animals that have no business being in the water column at all. That's unambiguous. Nothing about a Greenland shark suggests it climbed onto the tundra. Those animals fell in, or drowned, or were washed out, and the shark found them dead. Look at what that proves. It proves the shark scavenges. Confirmed. On the record. Now look at what it doesn't prove. It doesn't prove the shark only scavenges. A reindeer in the stomach tells you this animal will eat something dead. It tells you nothing about what it does when it encounters something alive. Both things can be true at once, and that's the part people flatten out. Notice how convenient the flattening is, though. Pure scavenger is a comfortable story. It keeps the slow shark harmless, keeps the Arctic tidy, and means nothing down there is taking anything you'd recognize as a mammal. The evidence doesn't support that comfort. It doesn't refute it either. It just sits in a stomach, refusing to say. But we still have the problem. If some of those seals were taken alive, the physics has to work. And the physics is brutal. You cannot chase down something 4 times your speed. That's not a difficult hunt. That's an impossible one. So the hunt doesn't happen while the seal is awake. Here's the thing about seals that changes everything. They're mammals, they breathe air, they sleep. And they cannot do those things on land in most of the Arctic because the Arctic is largely ocean, and the ice is unreliable. And a polar bear is very good at killing a seal that's sleeping on top of it. So seals sleep in the water. They do it in different ways depending on the species and the situation. Hanging vertically below the surface, drifting, or letting themselves sink slowly and then rising to breathe without fully waking. It's not the deep unconsciousness you experience, but it is a real reduction in vigilance, and it happens in open water, in the dark, for hours every day. That is a seal with its guard down. Not on the ice, where the bears are. In the water, where the shark is. Now run the shark's sensory stack against a sleeping seal. Smell finds it from kilometers out. The animal orients into the current and starts the long transit. Hours of it. Black water. No confirmation. Exactly the kind of patient bet that only a body running on nearly nothing can afford. The lateral line picks up the disturbance in the last stretch. And in the final meter, when there is nothing to see and the seal is inert, the ampullae read its heartbeat directly. Because being alive is an electrical event and you cannot switch it off. The shark arrives with no sound, no light, and no speed to give it away. And here's the reversal that makes the whole thing work. The slowness isn't a handicap in this approach, it's the weapon. Think about what a seal is watching for. It has been shaped by predators that announce themselves. The sudden acceleration, the pressure wave of a body committing to a strike. That's the signal a seal's entire nervous system is tuned to catch. A Greenland shark produces none of it. It doesn't accelerate, it doesn't rush. It coasts in on a 7-second tail stroke, displacing almost no water. A slab of cold meat drifting through cold water at the speed of a slow walk. It doesn't trip the alarm because it isn't doing the thing the alarm is looking for. Every fast predator in that ocean has an approach problem. How do you close the last 20 meters before the prey notices? The Greenland shark doesn't have an approach problem. It never crosses the threshold that gets noticed. The seal's defenses aren't beaten. They're bypassed. The thing that would save it from an orca is exactly the thing that doesn't fire. And the strike doesn't need speed either. Not much. The gap between a shark that's already touching you and a shark that has your flipper is centimeters. You don't need to be fast across centimeters. You need to be fast across the last 20 meters, and it never spent time in that zone at all. That's the hypothesis. And I want to be precise about the word because this is where a lot of this story gets sold as more than it is. Nobody has watched it. Not once. There is no footage of a Greenland shark taking a live seal. There is no observation, no recording, no eyewitness account from a submersible. What exists is an inference, built from what's in the stomachs, from the fact that seals sleep in water, from a sensory system that works in the dark, and from the physical logic that says a slow approach doesn't trigger a fast animal's alarm. That is a good inference. It's coherent, it's grounded in real biology, and it explains the evidence. It is not a fact. It is a hypothesis that fits, and it stays a hypothesis until somebody sees it. And now watch the whole thing close. The liver fishery. Thousands of animals killed over decades. And at the end, we couldn't say how they hunt. That felt like an oversight then. Like somebody should have been paying attention. It wasn't an oversight. It's structural. And here's the proof. A stomach is a list of nouns. Seal, reindeer, cod, squid. It is a perfect, high-resolution record of what went in, and it contains exactly zero information about how. The verb is not preserved. Cut open 10,000 of these animals, and you will have 10,000 lists of nouns, and you will still not know whether a single one of them ever killed anything, because the difference between hunting and scavenging leaves no trace in the tissue. It's a question about time, and a corpse has no time in it. We spent a century collecting the wrong kind of evidence, not because anyone was foolish, because it was the only kind available. So this is where the Greenland shark actually sits. We have a slow, half-blind, cold-running animal with the fastest prey in the ocean inside it, and a plausible mechanism for how it got there. And no way to confirm it that doesn't involve sitting in the Arctic dark, in the right place, at the right time, watching something that might happen once in a while, over a lifespan of 300 years. The most likely explanation for the seals is also the least observed predatory behavior of any large shark on Earth. And notice what just happened to the animal. It started as a sluggish garbage disposal. It is not that. It's something worse. A predator that solved the approach problem by refusing to have one, that turned a metabolic surrender into a hunting method, that gets close enough to touch you specifically because it isn't trying. The scary thing about this shark is not that it's fast, it's that it never needs to be. But that only gets it to the seal. It gets a mouth against warm skin in the dark. What happens next is a mechanical problem, because a seal that wakes up mid-bite is going to fight, and it's stronger and faster than the thing holding it. So the contact has to be decisive. The jaw has to do something that doesn't require a second attempt. And that jaw is not built like any shark jaw you're picturing. Open the mouth, and the first thing that's wrong is the size of it. You're expecting a great white's jaw, that huge triangular gape, the wall of serrated blades, the mouth that opens wider than a person, is tall. That's the shark mouth in your head. It's a mouth built to remove a large piece of something in one pass. The Greenland shark's mouth is small, short, a blunt transverse slot across the underside of a snout that doesn't taper into anything. It looks less like a weapon than like a mistake, like somebody put a cod's mouth on a body 4 meters long. Now look inside, and look at the two rows separately, because they are not the same tooth. They're not variations on a theme. They are two completely different tools that happen to be in the same head. The upper teeth are thin, narrow, pointed, spike-like, set in rows and pointing more or less straight down. There's no cutting edge worth mentioning on them. They're not serrated, they're not broad, they're not shaped to slice anything. They're pins. The lower teeth are the opposite. Broad, squat, each one carrying a cusp that leans over to the side, and here's the part that matters. They interlock. Tooth against tooth, edge to edge, across the whole lower jaw, so that what you have is not a row of individual teeth at all. It's a single continuous blade made out of teeth, laid across the bottom of the mouth like a saw with the handle removed. 2 rows, one that stabs and can't cut, one that cuts and can't stab. That asymmetry is the whole animal, and it tells you exactly what this bite is for, if you're willing to think about what those two tools do when they work together. They don't close on each other. That's the mistake. You're imagining a scissor. Top meets bottom, flesh in between, snip. That's how your teeth work, and it's how a white shark's teeth work, and it is not what this is. The uppers go in and stop moving. They're anchors. They pin the mouth to the surface of the prey, and now the shark is attached. Not holding, attached, hooked into the tissue by a row of spikes that are exceptionally good at not coming out. Then the body turns. The shark rolls. The head twists against the anchor point, and that continuous lower blade sweeps through the flesh in an arc, while the uppers hold the center of the circle in place. It isn't a bite. It's a rotation around a fixed point. And what comes out is not a torn chunk, it's a plug. A clean cylindrical core of tissue cut out of the animal like a sample taken with a hole saw. That's a corer, not a jaw. And once you see it, you cannot unsee what the rest of the body is doing, because now the slowness stops being a limitation on the attack and becomes a component of it. Think about what a white shark's strike requires. Speed. Enormous speed. Converted into force, because that mouth has to arrive and close, and remove, and depart in under a second. The prey is going to fight, so the entire transaction has to be finished before the fight starts. Every part of that animal, the warm muscle, the heat exchangers, the hydrodynamic body, exists to fund one violent moment. The Greenland shark has none of that. Zero. It cannot generate a strike like that at 1 degree Celsius with a tail that beats once every 7 seconds. If it needed speed to feed, it would have starved out of existence a long time ago. It doesn't need speed. It needs to not let go. That's the reversal. And it's clean. Once those upper spikes are set, the shark's own body mass and the roll do the work, and neither of those requires acceleration. A 4-meter animal turning slowly is still 4 meters of animal turning. The force comes from mass and leverage, not from velocity. It's the difference between a punch and a car being pushed into a wall. One is fast, the other doesn't have to be. You can see the aftermath. Circular crater wounds turn up on marine mammals in Greenland shark waters. Clean-edged, round, the exact geometry you'd expect from a rotating cut around an anchor point. They've been attributed to this species, and the shape of the wound matches the shape of the tool. Attributed, not filmed. Nobody has watched a Greenland shark make one of those wounds on a living animal. The tooth morphology says corer. The wounds say corer. The inference is strong, and the direct observation does not exist. And I'm going to keep saying that because it keeps being true about this animal. There is one place we can check the logic, though, and it's a small shark with a stupid name. The cookie-cutter shark is about the length of your forearm. It has the same architecture. Narrow spike-like uppers, a single interlocking blade across the bottom. And it does exactly what I just described to animals hundreds of times its size. Tuna, dolphins, whales. It attaches, it rotates, and it removes a perfect circular plug of flesh and leaves. The wounds are documented across the world's oceans. Nuclear submarines have come back with the rubber on their sonar domes cored out by them. So this isn't a hypothesis about how a jaw might work. It's an architecture with a known output. And the Greenland shark is running the same architecture at 10 times the scale, which raises an uncomfortable question about the target. The cookie cutter takes a bite and leaves. It doesn't need the animal dead. It just needs a mouthful. A Greenland shark taking a plug out of something isn't necessarily killing it either. Some of what we call feeding on marine mammals might be exactly that. A slow shark removing a piece of a larger animal and letting the rest swim off. We don't know which. Both leave the same evidence. Now the part that undercuts everything I just built? Watch a Greenland shark actually take bait on camera, and you may not see a roll at all. What you see is a mouth opening and the food going in, pulled, not cut. Because this animal has a large throat cavity and the ability to expand it fast, and that generates suction. A pressure drop. Water rushes in to fill it and carries whatever's floating nearby along with it. Suction feeding is real and it's common. A huge amount of the fish world eats this way. And here's why it fits this animal better than the corer does. Suction is cheap. There's no chase, there's no strike. You get close, you open. The pressure differential does the work. So the mouth might be 2 systems, not 1. A vacuum for anything small enough to inhale. Fish, squid, pieces of carcass. All the ordinary business of eating in a place where meals are rare. And the corer for anything too large, where you can't swallow it whole and have to remove a portion instead, which means the jaw isn't a specialist. It's a general solution to a specific problem. You are enormous, you are slow, and you have no idea what your next meal is going to be. It might be a piece of a dead whale. It might be a fish. It might occasionally be a sleeping seal. You cannot afford a mouth that only handles one of those because the ocean you live in serves whatever it serves, and you will not get to choose. So the answer to the seal question closes here, and it closes without any drama in it. The approach is stillness. The shark gets close, because it never does the thing that triggers the alarm. The contact is anchoring. The uppers go in, and the seal is now attached to a slab of cold meat 4 meters long. And what follows doesn't require a fight to be won, because a rolling body doesn't need to overpower anything. It only needs to keep turning. The seal is faster. That was never in question. Speed is for getting away from things you notice. And that's the feeding solved more or less. The search, the approach, the contact, the cut. A complete predator, assembled out of parts that individually look like failures. But here's what should be bothering you. Everything so far is about what the shark does to other bodies. None of it explains what's happening inside its own. Because the tissue of a Greenland shark is chemically hostile. Eat it fresh, and it will make you sick. Genuinely, seriously sick. There are accounts of sled dogs poisoned by it, staggering, unable to stand. Whole Icelandic food traditions exist purely as a workaround, a months-long process of burying and drying meat that would otherwise be inedible. The obvious read is that it's a defense. Something evolved to make this animal not worth eating. It isn't. It's got nothing to do with predators at all. Cut a piece off a fresh Greenland shark, cook it properly, eat it, and you will be in trouble within the hour. Not food poisoning trouble. Something else. The accounts describe staggering, loss of coordination, vomiting, a kind of drunkenness that isn't drunkenness. People unable to walk in a straight line, sled dogs going down in their traces, unable to stand. The Icelandic word for it translates roughly to shark drunk. It's not a metaphor anybody invented for fun. It's a description of what happens. So the obvious conclusion writes itself. The shark is venomous. Or poisonous, at least. It has evolved to be toxic, the way a pufferfish is toxic, the way a poison frog is toxic. Chemistry as armor. Don't eat me. That explanation is wrong, and the way it's wrong tells you more about this animal than anything else in its body. Start with the question nobody asks. Who exactly is this defense aimed at? A defense is a response to a threat. Poison frogs are toxic because birds eat frogs. Pufferfish are toxic because things eat pufferfish. There's a predator on the other end of every chemical defense in nature. That's what makes it a defense rather than a coincidence. Now name the animal that eats an adult Greenland shark. There isn't one. It's 4 to 5 meters long. It lives in Arctic water hundreds of meters down, and nothing in that environment is hunting it. It has no predator to deter. Building an expensive chemical defense against a threat that doesn't exist would be exactly the kind of unpaid bill this animal has spent 3 centuries refusing. There's a second problem, worse than the first. The toxicity isn't a specialty. It's not concentrated in glands, or in the skin, or in some organ set aside for the purpose. It's in the meat, all of it. The muscle, the tissue, the working substance of the body. Whatever's causing this isn't a weapon the shark built. It's what the shark is made of. So what is it made of? 2 compounds, and neither one has anything to do with defense. The first is urea. You have urea in you right now. It's the waste product of breaking down protein, and your kidneys are working continuously to get rid of it, because at high concentrations it damages proteins. Urea unfolds them. It's a destabilizer. That's why you urinate. to keep the levels down. A Greenland shark doesn't get rid of it. It hoards it. The concentration of urea in shark tissue is enormous, deliberately maintained, held in the blood and the muscle at levels that would kill you. Why would anything do that? Because of a problem you've never had to think about, which is that seawater is trying to dry you out. Here's the physics. Water moves across membranes toward whatever side is saltier. That's osmosis. And it's not optional. It's just what water does. A fish in the ocean is a bag of relatively fresh fluid sitting in a very salty solution, which means water is constantly being pulled out of its body, through the gills, into the sea. A marine fish is drowning in reverse. It's dehydrating while completely submerged, permanently, its whole life. Bony fish fight it. They drink seawater constantly and run pumps in their gills to excrete the salt. And it costs them energy every second of every day for as long as they live. Sharks cheat. Instead of pumping salt out, they load their tissues with urea until their internal concentration matches the sea. Now the gradient is gone. Water isn't being pulled out anymore because there's no difference in concentration for it to move down. The problem doesn't get solved. It gets deleted. It costs almost nothing. You're not running a pump. You're just declining to excrete something you were going to make anyway. The waste product becomes the solution. It's the cheapest possible answer to one of the most expensive problems in marine life, and for an animal that has built its entire existence around refusing to pay for things. This is not a coincidence. It's the same accountant. But you can't do that for free because urea wrecks proteins. Fill your body with it, and your enzymes start coming apart. So the trick would be self-defeating on its own, which is why it comes with a second compound, TMAO, trimethylamine N-oxide. And what TMAO does is stabilize proteins. It holds them in their folded shape. And it happens to counteract urea specifically, pushing back against exactly the kind of damage urea does. Sharks carry both in roughly balanced amounts, so the urea does its osmotic job while the TMAO keeps the machinery from falling apart. That's the system. Urea to match the ocean. TMAO to survive the urea. Every shark on Earth runs some version of it, and it's old. Older than the Arctic, older than the ocean this particular animal swims in. Sharks worked this out hundreds of millions of years ago and have never meaningfully revised it, which is usually what happens when something is right the first time. The Greenland shark didn't invent the trick. It inherited it, then pushed it harder than any relative because the place it went demanded more. Every shark is faintly poisonous, in other words. This one is only extreme about it. Now here's where the Greenland shark diverges, and it's the second half of the chemistry. Deep-sea animals carry more TMAO than shallow ones. It scales with depth, and it's one of the more reliable patterns in marine biology. Go deeper. Find more TMAO in the tissue. The reason is pressure. At depth, pressure physically deforms proteins, squeezes them out of shape, and TMAO resists it. It's a molecular brace. The deeper you live, the more bracing you need. Cold does something similar. Chill a protein and it becomes reluctant. The whole system stiffens, reactions slow, and stabilizing compounds help hold things functional. So take an animal that lives deep, in water at 1 degree permanently, for 300 years. Every environmental variable it faces is pushing the same direction. More depth, more pressure, more cold, more TMAO. And the Greenland shark carries a lot of it, along with a lot of urea, in flesh that is essentially soaked in the chemistry required to keep a body working under conditions that would shut a normal body down. Which brings us to what actually poisons you. The urea contributes. Enough of it in a meal will make anyone ill. But the real problem is the TMAO, and it's not even the TMAO's fault. When the tissue starts breaking down after death, bacteria convert TMAO into trimethylamine, and from there into related compounds that are genuinely neurotoxic. That's the staggering. That's the shark drunk. Read what that means? The shark isn't poisonous. The shark's corpse becomes poisonous. When the thing that kept it alive at depth rots into something that attacks nervous systems, the toxicity is not a trait. It's a decomposition product of the survival mechanism. So the whole framing inverts. This animal did not build a defense. It built plumbing. The cheapest possible osmotic solution, braced with the cheapest possible pressure and cold tolerance. And the poison is a byproduct that only exists after the animal is dead and no longer benefits from anything, which means it never evolved to hurt you. It cannot have. Whatever kills a sled dog is a consequence of chemistry that made perfect sense in a living shark at 400 meters in freezing water and stopped making sense the moment the animal was hauled onto ice. The toxin isn't a weapon. It's the receipt for the depth. There's a beautiful piece of evidence for that, which is the Icelandic tradition itself. The meat gets buried or pressed and left for weeks, then hung to dry for months. And at the end of that, it's edible. You cannot do that with a real toxin. You cannot bury a pufferfish for 3 months and eat it. Tetrodotoxin doesn't care how long you wait. But this washes out, breaks down, dissipates, given enough time. The fermentation isn't neutralizing a defense. It's waiting out a chemical imbalance in a body that was built for somewhere else. The shark's poison is temporary because it was never meant to be poisoned. And now look at what the chemistry just told us, because this is the part that opens the next problem. That TMAO load is a depth signature. It's a physical adaptation to pressure. The more of it an animal carries, the deeper it's built to operate. And this shark is loaded. We've been talking about it as an Arctic animal this whole time. Ice, cold, the far north. That's the image, that's the name, that's where every photograph comes from. The chemistry doesn't say Arctic. The chemistry says deep. Those are not the same word. And once you follow that, this animal turns up somewhere it has absolutely no business being. Look at where we've been putting this animal. Greenland shark. The name does the work before you've thought about it. You picture ice, a hole cut in it, a gray body coming up into flat white light. Every photograph you've seen was taken there. Every fisherman who ever talked about one was working the North Atlantic. The whole story so far has been an Arctic story. The chemistry disagrees. That TMAO load isn't a cold signature. Cold contributes, but the scaling that biologists have documented across the marine world is a depth scaling. The deeper an animal lives, the more of it in the tissue, because that's what braces proteins against pressure. And this shark is carrying a load that says something specific about where its body is built to operate. Not north, down. Those are different words, and we've been conflating them for the entire story, because in the Arctic, they happen to be the same thing. Here's the physical fact that breaks it apart. Cold water isn't a place. It's a layer. The ocean is stratified, warm at the surface where the sun hits, and it gets colder as you descend. And below a certain depth, it stops mattering what's happening above you. The deep ocean is cold everywhere. Not cool, cold. A few degrees, near freezing in places, and that's true under the ice, and it's true under the tropics. There is water at 4 degrees, sitting 2 kilometers beneath the equator right now, in permanent darkness. And it has been down there for centuries. The Arctic's peculiarity isn't that it's cold, it's that the cold reaches the surface. Everywhere else on Earth, you have to descend to find it. So what is a Greenland shark actually? Not an ice animal. A cold water animal. And the cold water habitat doesn't stop at the polar circle. It dives underneath the warm layer and keeps going south. A continuous mass of freezing water beneath everything. all the way to the equator and past it. The question stops being how far north does it live and becomes how far south can it go if it stays deep enough. The answer turns up in a place that has no business being in this story: the Caribbean, Belize. A deep water survey off the Belize coast. Researchers working a fishing hole in the Caribbean, dropping gear into deep water, and what came up on the line was a shark. Large, sluggish, and unmistakably a sleeper. A Greenland shark or something extremely close to it. Genetics couldn't fully resolve which, and that ambiguity matters, and I'll come back to it. But sit with the image first. Somewhere below the warm turquoise water in the postcards, in the black cold underneath the reefs and the tourists, a shark from the Arctic was swimming past, not migrating, not lost, living there, in a mass of cold water that has always been there, that just happens to sit beneath a sea we think of as tropical. That single record does something to the map. It doesn't extend the range a little. It suggests the animal's actual habitat has almost nothing to do with the geography we assigned it to. If a sleeper shark is in Belize, then the cold layer is a highway, and it runs the length of the Atlantic, and this species, or its immediate relatives, may be distributed through a volume of water we have essentially never surveyed for it. Now the uncertainty, and here it's load-bearing. There are other sleeper sharks. The Pacific sleeper is a recognized species, physically similar, running the same architecture in the North Pacific. There are others in the Southern Hemisphere, and the genetic boundaries between them are not clean. There's evidence of hybridization between Greenland sharks and Pacific sleepers, which is not what you'd expect from populations that have been isolated on opposite sides of the planet. Hybrids mean contact. Contact means these populations are not the sealed-off units the map implies. Think about what contact requires. For a Greenland shark and a Pacific sleeper to produce offspring, two animals from opposite ends of the planet had to be in the same water at the same time. There's only one way that happens: that the two oceans meet over the top through water cold enough for both of them, and the ice we picture as a wall is not a wall at all if you're already living underneath it. So the polar cap might not be the edge of this animal's world. It might be the corridor running through the middle of it. So the Belize animal might be a Greenland shark. It might be a related sleeper. It might be something in between, which is a category the taxonomy handles badly. And the honest position is that we do not know how many species of sleeper shark exist, where the boundaries between them lie, or whether the thing we call the Greenland shark is one animal with an enormous range, or several animals we can't currently tell apart. We named it after a place, and then found out it might live under the Caribbean, and might be interbreeding with something in the Pacific, and we still can't tell you how many of it there are. The name is a fossil of our own ignorance. Now go the other direction, because the vertical range is doing the same thing to us. They've been recorded shallow, right up under the ice, in a couple of 100 meters, in water so cold it's basically a freezer. And they've been recorded deep, well past 1,000 meters, into water that's still cold, but where the pressure has become genuinely serious. That's a vertical range of well over 1 kilometer, and the pressure difference between the top and bottom of it is enormous. Most animals can't do that. Pressure isn't like temperature. You can't just tough it out. It physically compresses proteins, and a body tuned for one depth generally has trouble at another. Deep-sea specialists are specialists precisely because that tuning is narrow. The Greenland shark seems to move through it freely, and the TMAO is why. Load your tissue with enough molecular bracing, and the pressure stops being a wall. It becomes a gradient you can drift up and down at will. The chemistry that makes the meat poisonous is the chemistry that makes the whole water column accessible. And that reframes the animal one more time. It's not living on a seafloor. It's not living at a depth. It's living inside a three-dimensional volume of cold water that spans a kilometer vertically and possibly an ocean horizontally, and it moves through that volume at 1 kilometer an hour with a nose that works across kilometers and no reason to ever be in a hurry, which is exactly why we can't count them. Think about what a population survey actually requires. You need to sample a defined area, count what you find, and scale it up. That works for a fish that lives on a bank, or a reef, or in a known depth band. You can draw a box around it. You cannot draw a box around this. The animal's habitat is a slab of cold water that may run from the Arctic to the tropics and from 200 meters to well past 1,000. And our sampling of it consists almost entirely of hooks dropped by boats fishing for something else in a narrow band of the North Atlantic, where humans happen to work. Every Greenland shark we have ever seen came from the part of its range that overlaps with our fishing industry. That's the sample. That's all of it, which is a sampling method with a name. It's called convenience sampling, and in most fields, it's grounds for throwing the study out. We are describing an entire species using only the individuals unlucky enough to be standing near us. Here's the thing that should unsettle you. We don't know if that's 10% of the population or a fraction of a percent. There is no way to know, because the rest of the volume has never been looked at with this animal in mind. The Belize shark didn't come from a Greenland shark survey. It came from someone fishing deep and being surprised. How many more of those surprises are down there? Nobody can say. That's not a rhetorical flourish. It's a literal statement about the state of the data. So the map we've been carrying this whole time is wrong in the most basic way. Not incomplete. Wrong. We named the animal after the one place we happened to meet it, then assumed the name described its life. The Greenland shark isn't from Greenland. It's from the cold, and the cold is most of the ocean by volume, and we have barely looked. But there's one place we have looked, and looked hard, and it's the last place you'd expect to find this animal's secrets. Not the ocean at all. A machine, in a lab, reading its DNA letter by letter. There's a lab freezer somewhere holding a piece of Greenland shark tissue, and in a real sense, we know more about what's inside it than we know about the animal it came from. That's not an exaggeration. We have the genome, the whole thing, the complete instruction set, sequenced, assembled, sitting in a database that anyone with a login can query. We can search it. We can compare it to a mouse, a human, a great white. We can ask it questions in the afternoon and have answers by evening. And we still don't know how long it's pregnant. That contrast is the whole thing. We solved the hard-looking problem, and the easy-looking one is untouched. And the reason is the reason it's always been. Reading a genome takes a week. Watching a life takes 300 years. One of those fits inside a career. Of course we sequenced it. It's the only move available. When you can't observe the animal, you interrogate the body, and DNA is the last piece of testimony a corpse has left to give. And immediately, the DNA is strange. The genome is enormous, something like double the size of a human's. Vastly more raw sequence in a shark than in you, which sounds like a compliment until you understand what genome size actually measures, because it isn't complexity. It never has been. There are amphibians running genomes many times the size of ours, and they are not doing more with them. What that extra material mostly is in the Greenland shark is repeat, transposable elements, mobile sequences that copy themselves and paste themselves back into the genome, over and over, accumulating like sediment. They're sometimes called jumping genes, and calling them that makes them sound purposeful. They're not. Most of them are doing nothing except making more of themselves. So the largest genome doesn't mean the most sophisticated animal. It means an animal whose genome has been colonized by its own parasitic sequence for a very long time and never bothered to clean house. which, if you've been paying attention, is exactly what you'd expect. This is the third time you've seen this pattern. The vertebrae never got mineralized because mineral costs energy. The eyes never got defended because defense costs energy. And the genome never got tidied because, well, editing is work. The accountant declined again. Look at what that gives you, though. A body that refuses to spend anything running for centuries, with a genome slowly filling up with junk because nothing in its life ever selected hard enough for a cleanup. And now the question everybody actually wants answered. Is the secret in there? Is there a longevity gene? This is where I have to be careful because this is where the coverage goes off a cliff. The story writes itself. 300-year shark DNA sequenced. Scientists find the code for immortality. You've seen that headline. It has been written many times. It isn't what happened. Here's what actually turns up in this kind of work. You take a genome from a long-lived animal, and you look at gene families associated with things that matter for aging: DNA repair, cell cycle control, tumor suppression. And you look for expansions, cases where a gene that most animals have 1 or 2 copies of appears in this animal 5 or 8 or 15 times. In the Greenland shark, there are expansions of that kind. Genes tied to DNA repair appear to be present in greater number than in comparable species. That's the finding. That is genuinely all of it. Notice what it isn't. It isn't a demonstration that those extra copies do anything. It isn't evidence that they're active, or expressed at meaningful levels, or that they confer any actual protection. It's a count. Someone looked at a sequence and found more copies of a thing than expected, and that is a starting point for research, not a conclusion. The step from this gene family is expanded to this is why the animal lives for 3 centuries is enormous, and it has not been taken. Expansions like that turn up in animals nobody finds remarkable. Gene families duplicate constantly. It's one of the most ordinary things a genome does with its time. Finding extra copies of a repair gene in a long-lived animal feels like a discovery, but you would find extra copies of something in almost any genome you stared at long enough. The real question is whether this expansion is the cause, or just one of 100,000 differences between any 2 species that happens to be the one matching the story we already wanted to tell. You would need cell cultures, functional assays, knockouts. You would need to actually test the machinery, and testing the machinery of an animal that grows a centimeter a year and can't be kept alive in a tank is a proposition nobody has a plan for. Same with the cancer story, which is even more seductive. The logic seems airtight. Cancer is a numbers game. Every cell division is a chance for a mutation, and enough mutations makes a tumor. Live 300 years at 4 meters long, and you have run more cell divisions than almost anything on Earth. You should be riddled with tumors. You should have died of cancer at 80, so it must have solved cancer, right? Maybe. Or maybe cold changes the arithmetic. Cell division isn't free running. It depends on metabolic rate, and this animal's metabolic rate is close to the floor. A body at 1 degree on almost no energy, growing a centimeter a year, is not dividing cells at anything like the rate a warm animal does. Fewer divisions means fewer chances. The shark might not have solved cancer at all. It might just be doing so little that it never accumulated the risk in the first place. Those 2 explanations, sophisticated genetic protection versus a metabolism too slow to break, are radically different, and the genome alone cannot distinguish between them. Anyone telling you it can is selling something. There's a harder problem underneath all of it, which is that we have no idea how a Greenland shark dies. Think about that. To study aging, you need to know what aging does. In humans, we know the heart, the arteries, the tumors, the cognition. There is a well-mapped set of failures. In this shark, that map is completely blank. We have never observed one die of old age. We have no idea whether their organs degrade, whether they simply keep growing until something structural gives out, or whether they're killed by something environmental long before any internal clock runs down. We are studying the genetics of longevity in an animal whose death we have never witnessed. Now stack the twists, because they're stacking fast. We sequence this animal before we ever saw one born. We can list its DNA repair genes, and we cannot tell you the length of its pregnancy. We have a searchable database of its complete genetic instruction set and no idea where its young spend their first 50 years. That is not a knowledge gap. That's a knowledge inversion. Everything about this species is backwards. The molecular is easy, and the obvious is impossible. And the reason is the same one from the beginning, wearing a lab coat now. A corpse gives you nouns. The genome is the biggest, most detailed noun we've ever pulled out of one. And it still isn't a verb. The sequence tells us what the animal is made of. It cannot tell us what the animal does. Here's the thing that should really land. The genome is a static object. It's the same in a newborn pup and in a 300-year-old adult. The letters don't change. So the one piece of evidence we have complete access to is precisely the piece that contains no information about time. We chose the tool that our timescale could handle. And in doing so, we chose the one tool that has nothing to say about theirs, which doesn't make it useless. It's a real tool, and it points at real questions. The DNA repair expansions are worth chasing, the transposon load is worth understanding, and there may be something in there eventually that matters for human medicine, but not yet. Right now, it's a list of leads, and the honest summary of what the Greenland shark genome has taught us about extreme longevity is: here are some places to look. That's it. That's the frontier. Worth saying plainly, because the distance between that and the headline is exactly where people stop trusting science reporting. Nobody found the secret. Somebody found a place to start looking. The looking will take decades, and it may end up saying nothing at all about us. And there's a cost to living this long that no genome will ever show you, because it doesn't happen in the DNA. It happens in the tissue, slowly, over centuries. The shark has been swimming through the ocean since before the Industrial Revolution, and the ocean has changed underneath it. Everything gets in. That's the part nobody thinks about when they hear 300 years. You imagine duration as a kind of prize. The animal wins, keeps going, outlasts everything. What you don't picture is that a body moving through water for 3 centuries is also a body being written on by that water, continuously, with no eraser. Start with the mechanism, because it isn't intuitive, and it drives everything that follows. Certain compounds don't dissolve in water. They dissolve in fat. Industrial chemicals, the persistent organic pollutants, the mercury compounds, the things manufacturing has been putting into the ocean since the 19th century. A lot of them are fat-soluble and chemically stubborn. They don't break down. They don't wash out. And when they enter a living body, they don't leave, because there's no exit. The body has plumbing for getting rid of water-soluble waste. It has essentially nothing for getting rid of something that has parked itself in fat, so it accumulates. In one animal, over its life, the concentration only goes up. That's bioaccumulation. Then it does the second thing, which is worse. A small fish eats plankton and accumulates a little. A bigger fish eats 100 of those small fish and takes on everything all 100 of them accumulated. A predator eats 100 of those. At every step up the food chain, the contaminant load concentrates, because you're not just eating a meal, you're eating the lifetime chemical history of everything in it. That's biomagnification, and it's why apex predators carry loads orders of magnitude higher than the water they swim in. Now put the Greenland shark into that equation. It's an apex predator, so it's at the top of the magnification chain. It's got that enormous oil-rich liver, which is exactly the kind of fatty tissue these compounds go looking for. And it lives for 3 centuries. 3 centuries of accumulation in a top predator. in a body built around a giant reservoir of fat. There is no animal on Earth better designed to collect industrial chemistry. It's not vulnerable to bioaccumulation. It's an instrument for it. There's a second reservoir nobody thinks about, which is the mother. In animals that carry their young internally and feed them on yolk, whatever is dissolved in the mother's fat has a route straight into the offspring. That isn't inheritance in the genetic sense. It's transfer. A chemical dowry, handed over at the start of a life that hasn't done anything yet. Whether it happens here, I can't tell you. Nobody has examined a pregnant Greenland shark for it because we've barely examined pregnant Greenland sharks at all. But the mechanism is well established across marine predators, and there's no obvious reason this animal would be exempt, which leaves a question with no answer underneath it. A pup born this year might be starting its 3 centuries, already carrying a share of what its mother spent 200 years collecting. I want to be honest about the state of the evidence here, because this is exactly the kind of claim that gets inflated. Contaminants have been found in Greenland shark tissue. That's established. It's been sampled. What I'm not going to do is give you a number, because the sampling is limited and the figures vary, and quoting a precise concentration would be pretending to a certainty that doesn't exist. What's solid is the principle and the direction. The mechanism is real, the animal is positioned perfectly for it, and the compounds are in there. Now here's the twist that makes this worth your attention. That old shark, the one with the estimate somewhere between 272 and 512 years, was alive before industrial chemistry existed. It was swimming in an ocean that had never contained a synthetic organic compound. Not fewer of them. 0. Those molecules had not been invented. And then, sometime in the middle of its life, humans started making them. And the shark just kept swimming. And the ocean filled up around it. And the compounds went into the plankton and up the chain, into the fish and into the seals and into it. It didn't do anything wrong. It didn't go anywhere. It stayed exactly where it had always been, moving at 1 kilometer an hour, and the world changed underneath it. Look at what that makes the animal. A single Greenland shark is a continuous chemical record of the North Atlantic across the entire industrial era. Its tissue contains a running account of what we put in the water, absorbed by a body that was already old when we started. The bomb pulse told us when it was born. The pollutants tell us what it lived through. Both of them are human signatures in a wild animal in the dark, and neither of them was ever supposed to be there. We used our own contamination to date it, and then we contaminated it again. But the chemistry is only half of what accumulates. Look at the outside. Photographs of these animals show bodies that have taken a beating, scarring, gouges, marks whose origins are frequently unclear, some of it consistent with mating, if the male-biting female inference from earlier holds, some of it from encounters we can't identify. And the coat pods on the eyes, generally on both, generally established, generally having been there a very long time. A young Greenland shark is presumably clean, unmarked skin, clear eyes, a body that hasn't met anything yet. Nobody has ever seen one. That's not rhetoric. The juveniles are absent from the record. The animals we pull up are the ones that have already lived through most of it. And what we call a Greenland shark is not really a species. It's a snapshot of a species at the end of a very long exposure. Every individual we've studied is damaged, and we have no baseline to compare it to. Think about what that does to every measurement we've ever made. When we describe this shark, we are describing the survivors of an extremely long exposure and calling it the species. It's like deciding what humans look like by only ever meeting people over 90. You'd get a real answer. It would also be wrong about nearly everything. Now the part that should genuinely bother you, and it's the one that goes back to the beginning. The eye, the lens, is the archive, the tissue that forms before birth and never turns over. That's why the radiocarbon works. It's a sealed capsule of original material. carried around unchanged for centuries, and it's the only clock this animal has. And a crustacean has been feeding on it for most of that time. We got our answer out of that lens. But think about what a lens that's been fed on for 200 years actually is. The parasite damages the cornea, the surface, and the nucleus is deeper. And there's no established finding that the copepod compromises the radiocarbon signal itself. I'm not going to claim it does, but the picture is unavoidable. The one piece of evidence that told us how old this animal is was hosting something that was consuming it the entire time we needed it intact. The clock survived. Barely. And the thing eating it was another consequence of the same longevity that made the clock worth reading. Everything that makes this animal remarkable also makes it accumulate damage. Every one of those unpaid metabolic bills. No warmth, no mineralized skeleton, no defended eyes, no tidied genome. Bought duration, and duration is exposure. The two are the same thing. You cannot have 300 years of life without 300 years of everything the world does to you. So the longevity isn't a prize, it's a ledger. And it only runs one direction. Every year adds a line. Nothing gets crossed off. Which brings us to the last twist, and it's the one that reframes the entire relationship between us and this animal. We keep treating age as an achievement, as if the shark accomplished something. It didn't. It didn't decide to live for centuries. It stumbled into a strategy of extreme cheapness in an environment where nothing killed it, and duration was the residue. The strategy has exactly one requirement. Nothing kills you. And for the entire evolutionary history of this species that held, the Arctic Deep was a place where a 4-meter shark could reliably not die. That's the only reason any of this exists. Then, sometime in the 19th century, a line came down through the ice with a hook on it. The shark had no adaptation for that. Nothing in 300 million years of shark evolution prepared a slow, cold, patient animal for a threat that arrives from above, kills instantly, and doesn't care how long you took to get there. Its entire strategy is a bet on nothing killing it. And something started killing it, at a rate its biology has no mechanism to answer. We are the failure mode. Not a threat among threats. The one thing the whole design cannot absorb. And the fishery isn't the past. They're still coming up right now in nets that aren't even meant for them. Right now, somewhere in the North Atlantic, a halibut boat is hauling a long line. And on that line, among the fish it went out for, there's a Greenland shark. Nobody wanted it. Nobody was fishing for it. The liver oil trade is over. That market died decades ago when petroleum and synthetics made shark oil pointless. The commercial hunt for this animal, the one that ran for a century, effectively ended. And they're still dying, in numbers nobody can put a figure on, because the thing that kills them now isn't a fishery. It's an accident. That's bycatch. The animals that come up on gear set for something else. It's one of the largest sources of mortality in the ocean for a huge range of species, and it's almost entirely invisible because there's no market for it, no landing record, no economic reason to count it. A shark comes up, it's cut loose or discarded, and the event leaves no trace in any database that matters. The Greenland shark takes bait. Of course it does. It's a scavenger with a nose tuned to find dead things across kilometers of black water. And a long line is a row of dead things hanging in the exact water it lives in. We built a machine for attracting the animal, and then we act surprised when it arrives. So the shark, that spent 3 centuries perfecting the art of finding a free meal in a place with no meals, finds one. Now do the arithmetic that this whole story has been building toward. An adult Greenland shark comes up. Say it's 200 years old, which is unremarkable for this species. That animal spent roughly 150 years as a juvenile before it could reproduce at all. It has maybe 50 years of reproductive life behind it. If the litter figure is anywhere near right, And remember, that number rests on almost nothing. It might have produced a handful of litters in that time. Might. It dies on the deck in an afternoon. To replace it, the ocean needs to produce a pup, and then that pup needs to survive for a century and a half at a centimeter a year, and then reproduce. The replacement time for one individual is longer than the entire history of industrial fishing. That's not a slow recovery. That's not a population under pressure. That's a species doing arithmetic it cannot win, because the removal takes an afternoon and the replacement takes 150 years. And there is no gear in the biology to close that gap. No compensatory burst. No faster breeding under stress. Nothing. Every haul that brings up a Greenland shark subtracts centuries from a standing stock that took centuries to build. And it gets worse, because of the thing you can't see on the deck. You cannot tell whether it's mature. I mentioned this before, and it deserves the weight. A 100-year-old juvenile and a 200-year-old adult are both large gray sharks. There's no visual marker. Maturity in this species is not written on the outside, and it can only be confirmed by examining the reproductive organs, which requires the animal to be dead and cut open, which is not something a halibut crew is going to do at 3 in the morning. So the fishery cannot distinguish between an animal that has reproduced and an animal that spent a century getting ready to and never got the chance. Every one of the second kind is a total loss. 100 years of biological investment, surviving the cold, the scarcity, the parasites, the dark, canceled with a genetic contribution of exactly 0. That's what the liver fishery did thousands of times, for decades, and nobody knew because the maturity number didn't exist until long after the boats stopped. We were harvesting a species' future, and we couldn't see it. Not because anyone was careless, because the information required to see it took another 50 years and a nuclear weapons program to produce. Now here's the part that makes this genuinely different from every other conservation story you've heard. Normally, this is where someone tells you the population estimate, how many are left, whether it's declining, and how fast. That number is the entire basis of fisheries management. You count the stock, you calculate what it can sustain, you set a limit. There is no number, not a bad number, not a contested number, nothing. Nobody knows how many Greenland sharks exist. Not to the nearest 1,000, not to the nearest order of magnitude. Any figure you see quoted is somebody's arm waving. That is a genuinely unusual place to be, with a large vertebrate in this century. We have estimates for tigers. We have estimates for blue whales, which range across every ocean on the planet. We have estimates for animals nobody ever farmed, hunted, or had the slightest economic reason to care about. Counting things is the oldest job in conservation biology, and it has counted almost everything big enough to see. This one, no. And by now you know exactly why, because every reason is something we've already walked through. You can't count them because you can't survey their habitat. It's a slab of cold water that may run from the Arctic to the Caribbean and from 200 meters to well past 1,000. And our sampling of it is a scatter of hooks dropped by boats fishing for something else in the narrow band where humans happen to work. You can't count them because you can't find the young. 50 years of the growth curve is missing from every dataset we have. There's no nursery. Any population model needs recruitment. How many new animals are entering the population? And for this species, that input is a blank. You can't count them because you don't know what a healthy population looks like. There's no baseline. The liver fishery ran for decades before anyone was recording anything useful. So whatever we're looking at now is the aftermath of a hunt we never measured, and we have no idea what came before it. And you can't model them because the model needs a reproductive rate. How often does a female breed? Nobody knows. That single unknown makes the entire calculation impossible. You cannot project a population forward without knowing how fast it makes more of itself. So the situation is this. We have a species with the slowest replacement rate of any vertebrate on Earth, dying in unknown numbers, from an unrecorded source, with no population estimate, no baseline, no recruitment data, and no reproductive rate. The IUCN lists it as vulnerable. That's the honest position. It's what you say when the biology screams danger and the data can't confirm it. It's a listing assembled entirely out of arithmetic. No survey underneath it. No trend line. A life history and a calculator. Read that carefully. That listing isn't based on a measured decline. It's based on what we know about the animal's life history. The maturity age, the growth rate, the longevity, and what that arithmetic implies about its ability to absorb mortality. The conclusion is an inference from the biology, not an observation of the population. We're not watching this species decline. We're calculating that it must be and unable to check, and that's the reversal that closes this whole thing because we've been treating the not knowing as a curiosity this entire time, a charming mystery. A strange gap in the literature about a weird Arctic fish. It isn't charming, it's the actual danger. If you can't count them, you can't manage them. If you can't detect a decline, you can't respond to one. If a population is dropping right now, the first evidence would take decades to accumulate. And by then, the animals that would have rebuilt it would already be gone, because they take a century and a half to arrive. The mystery isn't a story we tell about this shark. It's the mechanism by which it could disappear without anyone noticing. That's the trap. A species that vanishes slowly enough is a species that never triggers an alarm. There's no crash, no collapse, no year where the catch drops and everyone panics. Just fewer of them. Gradually, over generations, in a range nobody surveys, at a rate nobody measures, until eventually the boats stop bringing them up and nobody can say when it happened. You cannot lose a cod population that way. Cod crash loudly. The Greenland shark would go quietly, and underneath all of it, under the bycatch, the missing nursery, the absent population estimate, the unknowable reproductive rate, sits the same fact that has been sitting there since the first page. Everything about this animal is unknown for one reason. One, it's not the depth. It's not the cold. It's not the remoteness or the ice or the expense. It's that we are watching an animal that lives for 300 years using tools that last a few, wielded by people who live 80. We're not failing to see it. We're failing to see it for long enough. Go back to the beam. A cone of light in black water, maybe 10 meters of visibility, everything outside it absolute. That's the entire instrument. That's what we send down. A bright circle in an ocean that is mostly not bright circles, held there by a cable, burning power, on a clock, and a gray shape crosses it, slow enough that the camera operator isn't sure it's alive. 4 meters of animal, eyes fouled, skin scarred, passing through the light at a walking pace, and then the tail sweeps once and it's gone. That's the encounter. That's what we get. Now put a number on it. Say that shark is 250 years old. Unremarkable, middle of the range. Nothing special. And say the ROV had it in frame for 30 seconds. 30 seconds out of 250 years. Do the division and it stops being a metaphor. That's a fraction so small it doesn't have a useful name. It's less than a heartbeat in a human life. It's not a glimpse. It's not a sample. It is, statistically, nothing. A single frame pulled from a film that has been running since before the camera was invented and will keep running long after the cable is coiled and the ship has gone home. We didn't observe that animal. We were briefly in the same place as it. And that is the whole answer. It's the thing this story has been circling from the first sentence, and it was never about the ocean. We've been telling ourselves its remoteness, That the Greenland shark is unknown because it lives somewhere hard, cold water, deep water, under ice, far north, expensive to reach. It's a lie, and a convenient one, because it makes the ignorance somebody else's problem, a funding problem, an engineering problem, a problem that a better submersible and a bigger grant would solve. We have been to the bottom of the deepest trench on the planet, repeatedly. We have filmed animals at hydrothermal vents in water hot enough to destroy equipment. We have followed sperm whales into the black. We have found the giant squid, which was a legend for centuries, in the open ocean, on camera. When humans decide to look at something in this ocean, we find it. Distance was never the barrier. Depth was never the barrier. The Arctic is cold and expensive, and it is not the reason. The reason is that we brought the wrong axis. Every tool we've built for studying life in the sea is a tool for capturing space. Where is it? How deep? How far? What's the range? Map the distribution. We are extraordinarily good at space. We can put a lens anywhere on this planet. And every one of those tools is nearly worthless against time. A dive lasts hours. A drop camera lasts days. A tag lasts months, maybe 2 years, before the battery dies or the animal sheds it. A funding cycle lasts 3. A scientist's whole career in the field lasts 40, and then they retire, and the students who take over start their own clock from 0. Stack every one of those against 300 years, and you have a stack of nothing. Our longest instrument is shorter than this animal's childhood. Money doesn't fix it. You can build a better tag. You cannot build a tag that outlives the person who attached it, gets maintained by strangers who never met her, and reports back in the year 2300, not because the engineering is impossible, but because no institution on this planet has ever kept a promise that long. That's not a gap in the data. That's a mismatch in dimension. We have been trying to measure duration with rulers. Look at what happened every single time we tried. We wanted its age. There were no growth rings because a skeleton too cheap to mineralize keeps no record. So we drilled into an eye, into a lens that formed before birth and never changed. And we got an answer only because a nuclear weapons program had accidentally date-stamped the atmosphere. We didn't measure that shark. We measured our own explosions, reflected back at us out of its eye. And even then, the answer came out as a range wider than the country most of you are listening from. We wanted its life history. We got a genome. Billions of letters, complete, searchable, done in a week, and the genome is static. Identical in a newborn and a 300-year-old. The one piece of evidence we can fully access is the one piece with no time in it at all. We wanted its behavior. We had a century of dead sharks, thousands of them, and every stomach was a list of nouns. Seal, reindeer, cod. Perfect resolution on what went in and zero information on how, because a corpse has no duration and the verb doesn't survive the animal. Every workaround led to the same place. We could only ever ask questions our own timescale could answer, and those questions had nothing to say about theirs. So the reversal is this, and it's been sitting under the whole thing. The Greenland shark isn't slow. We're fast. Absurdly, unusably fast. A species that matures in 13 years, works in 40, and dies in 80, trying to observe something that treats a century as adolescence. Put us next to that animal, and we're the strange ones. We're the flicker. A human lifetime, from birth to death, fits inside the childhood of a fish, with room left over. The shark isn't hiding in the Arctic. It's hiding in a time scale we can't stand still in, and that reframes the animal from the ground up. It's not a slow shark that happens to live a long time. It's a body that took everything: warmth, speed, a mineralized skeleton, functioning eyes, a tidy genome, every single thing another animal would have paid for, and traded all of it for duration. That was the whole design. Give up the moment. Buy the century. It didn't refuse those things because it was primitive. It refused them because in a place with no food, no light, and no predators, the animal that spends least lasts longest. The slowness isn't a symptom of the longevity. It's the price of it. Same transaction. Seen from either end. And the invisibility came free with the purchase. That's the part that should stay with you. Nothing about this animal evolved to hide from us. There's no camouflage in it, no evasion, no strategy directed at humans at all. It has no idea we exist. It simply lives on an axis our instruments don't extend along, and that has been enough. It's not concealed. It's out of range, which is why the danger is what it is. Because the thing that makes this animal unknowable is the same thing that makes it unsavable. Look at what we'd actually need. To protect a species, you have to notice it changing. And noticing requires two measurements far enough apart to differ. For a cod, that's a season. For a whale, a decade. For this animal, the interval that would show you anything is longer than the profession of the person doing the measuring. So the instrument that failed to find its nursery is the same instrument that would fail to find its collapse. It isn't two problems, it's one problem, pointed at two questions. And that's the sentence I'd like to leave sitting in the room. We are not going to lose the Greenland shark to malice or to appetite or to anything that would show up in a headline. We would lose it the way you lose a language, one speaker at a time, over generations, with nobody in the room old enough to hear the difference. So, last thing. There is an animal alive right now, in cold black water somewhere in the North Atlantic. Or maybe under the Caribbean. We're honestly not sure. That was born before anyone thought to ask any of this. Before the fishery. Before radiocarbon. Before the word biology meant what it means. It has never been caught. It has never been tagged. It has never been photographed. And it never will be. It is 3 meters long and it has not reproduced yet because it is still a child. It will be alive when you are not. It will be alive when everyone who works on this species has retired and when their students have retired. And it will still be adding a centimeter a year in the dark, carrying industrial chemistry it never asked for in a liver full of oil, with something eating its eyes. We will never know it. Not that individual, not any of them. Not really. Because knowing an animal means watching it live, and no human being will ever watch a Greenland shark live. The beam is still down there, a cone of light in a volume of black water, the size of a continent, on a cable, on a clock. And the shape moves through it once and keeps going.